Good morning, ladies and gentlemen. We will open our conference on wastewater-based surveillance infectious disease. And we have the pleasure to welcome our Vice President for Internationalization, Guillaume Fiquet, to open officially this colloquium.
So, thank you very much. So, ladies and gentlemen, distinguished guests, dear colleagues, I'm really delighted to welcome you today in the name of our President, Nathalie Drachtemam, to the Paris Brain Institute, which is part of Sorbonne University, for this international conference. My name is Guillaume Fiquet. I'm Vice President of the University in charge of international relations and also economic partnership. So this conference is really dedicated to a scientific field of major interest, epidemiology, and its role in public health research. So we know that Sorbonne University is an important player in epidemiology, and we've really tried to foster a multidisciplinary approach to this field. We have in science biology, environmental microbiology, statistical modeling, and biostatistics, applied mathematics, hydrology, medical research. We have virology, infectiology, clinical trials, but also in the humanities and social sciences. For example, the study of social determinants and the question of access to care. So we have also critical math in health, science, and the humanities, and we have been federating major research forces in these fields for several years. Since 2014, Sorbonne University has been home to the Institut Pierre-Louis d'Épidémiologie et de Santé Publique, for instance. So we are very proud of this established research carried out over a long term. Maybe that's the reason why we were able, to some extent, to respond to the health crisis caused by the COVID-19 pandemic. And this was also a pandemic that highlighted the importance of wastewater-based epidemiology as never before. We have one of our flagship program, OBEPINE, which is a prime example. Was strongly supported by university and ministerial policymakers. It had led to major advances, with nearly 40% of the French population that was monitored by March 2021. But this crisis reminds us the imperative need to take risk in order to finance fundamental research, because it starts there. It starts with research. And it also reminds us that support for research, but not be seen as just a windfall, but it must be a long-term effort. And I think that efforts have paid off. OBEPINE has now been rolled out nationwide, has been made permanent. It's also expanding into the monitoring of other high-risk pathogens. And the French General Secretariat for Investment has requested the development of a national research platform as well, to detect and monitor emerging infections. Of course, epidemiology is evolving. With the development of new indicators, we are now able to detect transmissible infection, HIV, sexually transmitted diseases, viral hepatitis, or chronic diseases, diverse inflammatory diseases, cardiovascular diseases, but also to study antibiotic resistance. And I think that these are all real major areas of concern. So I would say, of course, I said that this morning in front of a very important panel, that epidemiology is more essential than ever and more topical than ever at a time when viruses are re-emerging and others are making their appearance. So this conference gives me the opportunity, first of all, to thank all actors engaged here in Sorbonne University in that research, and also reaffirm that Sorbonne University is committed to the development of this research. I think that epidemiology is a formidable tool for public health. It's a crucial decision-making tool, enabling us to measure and prevent the health risks to which populations are exposed. Second, I think that the vocation as academics is also to advance knowledge for the benefits of society as a whole. The World Health Organization, whose representative will be there this afternoon, no longer defines health as a simple absence of disease. Rather, it's a more holistic concept of physical, mental, and social well-being. And yes, we need an integrated vision of healthcare. It is essential. It must be seen as a whole that transcends borders. It includes physical, environmental, sociological, and economic aspects. And I think that all these fields will probably be covered during this day. It also includes improved and equitable access to care and prevention. But this can only be achieved by increasingly pooling research and innovation strengths, by promoting interdisciplinary, participatory, and translational research. I'm sure our scientific community understands this and takes action every day in a lab, the advanced research, and they are supported here by university structure in close proximity to users. We have the service called Autonomia. We have the University of Patients. We have also education departments with a master in public health, for example. Then, within the framework of our international strategic partnership, our European Alliance 4-Year Plus, notably via the SWATH project, so you'll probably hear of that project today on wastewater-based epidemiology. And I could see a lot of colleagues from the University of Geneva, Professor Antoine Flau. We have colleagues from Fiocruz, not maybe colleagues from IIT Delhi, India, or UNAM in Mexico. But I see indeed a very international panel this morning. And finally, within our Sorbonne University Alliance here in Paris, with the creation of a multidisciplinary institute dedicated to global health, which is really designed to bring together all these vital forces. And indeed, this institute will draw on research in epidemiology, cancer, nutrition, autonomy, and work will continue in the fields of biomedical humanities and health economics. So, to conclude, public health is a really global challenge. We are certain that a collective response is required here. It's challenges for all of us, for humanity, and we must face it. All of us here today, we share a common goal to advance scientific research to enable governments to have a clear vision of the health risks to which their population are exposed, and give them the means to take action against disparity in access to healthcare. So at this conference, I invite state representatives, intergovernmental agencies, European commissions, academics and researchers to reflect on the kind of research, support, and openness required to make this work enduring and sustainable. Let us champion the scientific progress our society needs and make this advancement accessible to all. This responsibility rests on each of us. Thank you very much, and I wish you a very exciting day around this topic. Thank you again.
Thank you, Guillaume. So now we are ready to start our first session on with the first topic, wastewater-based epidemiology for pathogens monitoring. And I will call our chair for this first topic, Bilge Arslan from the Turkish Water Institute.
Okay. I think we can first invite our speakers to the platform. Okay, please. Okay, ladies and gentlemen, I welcome all of you to the first session of the conference on wastewater-based surveillance on infectious diseases towards a global perspective, organized by World Health Organization, for EU Alliance, OBEPINE, Geneva Health Forum, Sorbonne University, and the Paris Panthéon-Assas University. I will start by introducing myself. My name is Bilge Alparslan Kocamemi. I am a professor in the Department of Environmental Engineering at Marmara University in Istanbul, Turkey. Since the start of COVID-19 pandemic, I am also working as the nationwide coordinator of wastewater surveillance of Turkey under Turkish Water Institute, which is a general directorate under Ministry of Agriculture and Forestry of Turkey. And on behalf of Turkish Water Institute, I play an active role in the global consortium for wastewater and environmental surveillance for public health, which is known as GLAVACON. And I'm co-leader of the training and capacity building working group of this consortium. And also I'm serving as the national contact point of Turkey at the GeoFrixe meetings on the wastewater and environmental surveillance with European Union member states as an observer country. Last but not least, I'm also the co-leader of the Water Quality and Health Task Force of the World Water Council, and I'm one of the directors of the International Water Resources Association. And today I am really honored to be here to moderate the session on wastewater-based epidemiology for pathogen monitoring. Although the use of wastewater surveillance to monitor public health is not a new concept and its historical background extends to the polio eradication, it received great attention with the COVID-19 pandemic. This is due to its power in monitoring infectious disease presence and spread in a community without distinguishing between asymptomatic and symptomatic individuals, using small number of samples in a very cost-effective way. And today, as of our knowledge, there are 72 countries in the world using this tool, not only to monitor SARS-CoV-2, but also for the other viruses like monkeypox, influenza, dengue fever, and the others. And additionally, over the past 4 years of the COVID-19 pandemic, wastewater-based epidemiology has been accepted as an early warning system. And the very recent application of the GLAVACON supersize, practicing the simultaneous collection and analysis of aircrafts and airports wastewater samples around the world, is a milestone towards the using of wastewater surveillance as an early warning system to protect public health. Through this conference platform, we will provide a comprehensive look at the value of wastewater-based epidemiology in monitoring a range of infectious diseases. And we will have an opportunity to listen to various case studies from our distinguished international experts and discuss the integration of wastewater-based epidemiology into national and international health monitoring strategies. And without further ado, I would like to start our session with our first speaker, Vincent Maréchal. Yes, Vincent Maréchal is a professor of virology at Sorbonne University. His research group mainly focuses on human oncogenic viruses and antiviral strategies. Since 2020, he has headed the OBEPINE network, which is an epidemiological observatory in wastewater, which tracked the COVID-19 epidemic in France between March 2020 and April 2022. He is currently in charge of the OBEPINE Plus project, a national research and development platform bringing together 20 academic and industrial partners to carry out research projects dedicated to techniques for detecting and monitoring emerging infectious diseases via wastewater analysis. Today, Professor Maréchal will present us the OBEPINE achievements during the COVID-19. Yes, Professor Maréchal, floor is yours. Okay, thank you.
Thank you to all of you. Thank you for this very nice introduction. So I will, yeah, everything is working. So as we just said, OBEPINE is the French research and development network that has been working very early during the COVID-19 crisis. As you know, COVID-19, of course, is a respiratory infections, but as a matter of fact, the virus is also highly replicating in gut, which makes possible very early to detect the virus in wastewater treatment plants. So the basis of OBEPINE project was to test an hypothesis, which is, can we infer incidence rate from wastewater viral load in France? So for that, we set up an interdisciplinary research consortium, and the founders of OBEPINE are on these slides, and I really want to tell how much it was a pleasure to work with them on this project. So people are working in virology, microbiology, environmental microbiology, mathematics as well. And I should say that OBEPINE is clearly based on two sides. The first one is to develop genomic approaches to detect SARS-CoV-2 and other viruses, but also to make a lot of mathematical modelization to try to smooth the data and to be able to make a good agreement between incidence rates that can be evaluated with massive testing in the population and what we can get in wastewaters. So basically, the first work has been done in France between March 2020, so at the very early beginning of the crisis in France, until March 2020. And as on these pictures, what you can see is the level of, yes, it works. The level of virus in one specific wastewater treatment plants around Paris. You can see here that the viral load is strikingly decreasing during the first lockdown, which was a strong and 6 weeks lockdown in France. So this was the proof of concept of the system, and based on that, the French government proposed us to develop a national monitoring system, including 200 wastewater treatment plants, which is more or less 40% of the French population. So basically what we could observe, and you can see on these pictures, on these examples, is that the incidence rate in black is closely related to the incidence level you can measure by wastewater-based epidemiology, provided that modeling and techniques are really up to date and efficient. So what we've done also is to try to monitor variants in wastewaters. You can do that by, for example, variant-specific quantification. As you can see here, it has been done by our colleagues of Haute-Paris, or as you can see on the right side, which has been done in Nice by Pascal Barbry team, which is an example of high-frequency sequencing in wastewater, and it has been done in Nice, in the south of France, from January 21 to June 24. So OBEPINE also has been involved in different questions. We investigated the infectivity of SARS-CoV-2 in stools, human stools and wastewaters, and provided evidence that infectivity is very poor. We also some work in Paris during the 2022 epidemics of monkeypox, and could provide evidence that you can detect the virus in sewers very early. And more recently, colleagues in Institut Pasteur Guyana have been able to show that and to detect circulating type 3 vaccine-derived poliovirus. So I would say that OBEPINE work have been expanding towards different pathogens, but also different techniques. I would conclude by saying that OBEPINE Plus, as we said previously, is a research and development platform that is supported by the French government, and it's included in the National Prevention Plan against emerging infections. It gathers more than 20 academic and industrial partners who wants to work together to develop tools to anticipate future epidemics. This tool will be transferred to the current monitoring network called SUMO in France. And it's also very important to underline that this network is designed also to promote wastewater-based epidemiology in countries with limited resources. And we will have some example of that kind of application in the coming talks. Thank you very much for your attention.
Okay, thank you, Professor Maraşan, for your insightful presentation on OBEPINE. Your valuable contributions have significantly enhanced our understanding of OBEPINE, which represents a best practice in developing a national platform for research and development of in research wastewater epidemiology. Congratulations to you and your team. Our next speaker is Dr. Milena Busova. She is an assistant professor at the Institute of Hygiene and Epidemiology of First Faculty of Medicine, Charles University, and General University Hospital in Prague. She is graduated in molecular biology and genetics. Her professional focus includes preventive medicine, epigenetic, and genotoxic substances from the environment, wastewater-based epidemiology, nutritional toxicology, and food safety. Currently, her research focus is on the prevention of damage to human health. Today, Dr. Busova will present us the relation between clinical data and wastewater parameters during COVID-19 pandemic in Czech Republic. Yes, Dr. Busova, the floor is yours.
Dear ladies and gentlemen, dear organizers, thank you so much for the opportunity to speak at this conference. Allow me shortly or briefly inform you about our research, which was focused on the presence of SARS-CoV-2 RNA in wastewaters during the COVID-19 pandemic in the Czech Republic. Wastewater-based epidemiology is a unique method which uses some parameters, chemical parameters or biomarkers from wastewaters for target assessment of lifestyle, including health and exposure of population to various substances. Previously was used for the monitoring various chemical substances, for example, metabolites of drugs, other pharmaceuticals or illicit drugs. Now is currently used for the detection of various biological agents, especially adenoviruses, noroviruses, including polioviruses. And at this moment, I would like to briefly mention situation with poliomyelitis. In 1949, poliovirus was successfully cultivated in human tissue because it was a very serious disease between a pediatric population. Focus about 1940 work and focus on the development of vaccines, effective vaccines. And Czechoslovakia was one of the 5 countries in the world when in 1957 to produce own effective vaccine against this serious disease. At this moment, I would like to mention the name of our famous and prominent virologist, Dimitri Slonim, which contributed to development of vaccine and contributed to successful vaccination between pediatric population. Czechoslovakia became, from 1960, the first country in the world being poliomyelitis free. And currently, polio occurs only in small endemic localities, Afghanistan and Pakistan. But risk of spread of polio exists, and since 2020, the Czech Republic continued to monitoring wastewaters for the presence of polioviruses. COVID-19 is a respiratory infection, communicable infection transmitted due to respiratory route, and advantage is it can be excreted, viruses, RNA can be present in wastewaters due to feces or urine. And advantage is for wastewater-based epidemiology is incidence in presymptomatic or asymptomatic persons which excreted virus RNA, especially when their respiratory specimens are negative. And from 2020 to 2022, our Institute of Hygiene and Epidemiology, First Faculty of Medicine, collaborated with Water Research Institute in Prague and on the project named COVMON. It was water monitoring used as an early warning tool for epidemic outbreak. This project was supported by Ministry of Interior and included into the security research. On this graph, you can see total numbers of people with current COVID-19 during this period. And common study was focused on the four wastewater treatment plants with different sizes, two large wastewater treatment plants, two little. Localization is on this map. One is bigger were from Prague and Brno city, and little from Kladno and Břeclav. And from these wastewater treatment plants, were taken samples for chemical and biological analysis. Yeah, parameters you can see here. And virus identification was provided due to real-time PCR. And numbers of active positive cases were acquired from the National Institute of Public Health in Prague. On this table, you can see predominant SARS-CoV-2 viruses variants during this period. All variants are here, Wild, Alpha, Delta, and Omicron. And trends of viruses RNA concentration is in this table marked by red line, numbers of active COVID-19 cases by blue line, hospitalized COVID-19 cases in green line, and these data very good correspond between correspond very good. And on this table you can see significance correlation. Significant correlations are marked bold by bold. Highest Spearman correlation coefficient were present in dark color in these tables. Yeah. And our research confirmed significant correlations between population status in the monitored area with the clinical data and virus RNA concentration. And strong correlation were found active and hospitalized cases and concentration of virus RNA in detected wastewater treatment plants. Our results reflect COVID-19 burden in our republic and local communities. And our research confirmed that municipal wastewater surveillance should be used for a prevention of early warning purposes. And the goal and confirmation, good idea is support the proposal Urban Wastewater Treatment Directive, this legislation, to introduce the coordinated systematic use of wastewaters and environmental surveillance as part of global infrastructure for preparedness of pandemic. I would like to thank you for your attention, and I would like to thank my dear colleagues, Milan Tuček, Petr Smejkal, Anna Horňáková, my colleagues from Institute of Hygiene and Epidemiology, and especially my dear colleague Věra Očenášková, sitting here from Water Research Institution, and to PhD student, medical doctors, Iris Kesha Nadio and Romana Pospíšilová, PhD candidates. Thank you very much for your attention.
Okay. Thank you, Dr. Busova, for your insightful presentation. It is highly valuable for us to hear strong correlation between case data and wastewater data in Czech Republic. Your remarks reinforces our global efforts for 4.5 years and demonstrates that the wastewater-based epidemiology approach can be used to predict the epidemic trends. Thank you. And we will continue with our third speaker, Nicholas Gressley. He is a professor in the Department for Infectious Disease Epidemiology and head of the Vaccine Epidemiology Research Group in the School of Public Health at Imperial College London. He works on vaccine trials and disease surveillance with a particular interest in novel sequence-based surveillance tools and data analysis methods. He collaborates with many partners worldwide to generate the evidence base for improved surveillance and disease prevention, particularly in low and middle-income countries. Today, Professor Grassley will present us the wastewater surveillance for pandemic prevention, mainly focusing on the learnings from polio and typhoid. Yes, floor is yours, Professor.
Thank you very much, Bilge. Do I need to change the slides here? Different
If you want. Yeah. We didn't have the screen, yeah.
We have to close, I think.
No,
First close it.
you know?
No, but I can estimate. It is here, I think. No. We have to close it and we have to open his presentation.
You have to switch on the screen. Yeah, but we just have the screen.
Presentation is yours. It's on the desk.
Yes, it was.
This is Mustafa, this is yours.
No.
Maybe we can enter
No.
Tuchel's folder.
Non.
Oui, on va laisser Diane.
Okay.
Excusez-moi, j'étais partie sur un autre sujet. Sorry.
Always happens. Okay, no problem. Okay, perfect.
On a switch, voilà. Tu mets sur mode présentation. Oh, Nick, sorry for that.
Okay. Okay, sorry for the interruption. Thank you for the invitation to speak, and thank you to you all for being here. And I think we have people online as well. Is that right? So hello to those who have joined online. Welcome to this session. So yeah, my name is Nick Grassley. I'm a professor of epidemiology at Imperial College London. And in this very brief presentation, I want to talk about wastewater surveillance for pandemic prevention and what we can learn from polio and typhoid. Okay, so coming from the UK and starting in London in 2022, we detected poliovirus circulation early in that year. That detection over time increased in abundance, and by sampling upstream from the initial site of detection, it was possible to locate where poliovirus was circulating in London. I don't know if you know London, but in the northeast in Hackney. And what this meant was that we could respond with a targeted catch-up immunization campaign, and eventually with a London-wide mass immunization campaign to children 1 to 9 years old. So by successful wastewater surveillance, what I mean here is where surveillance leads to public health action. So more broadly, of course, wastewater surveillance has been key to the global polio eradication program. Milena mentioned that the last two countries that remain endemic for wild-type polio are Pakistan and Afghanistan. Across the world, including in Pakistan and Afghanistan, wastewater surveillance has been used to detect circulation of poliovirus. In this map showing that detection through wastewater is much more widespread than the limited reporting of cases. And that's because infection with poliovirus only rarely leads to paralytic poliomyelitis that is reported. And so there's a clear value add of wastewater surveillance in detecting asymptomatic infection. And that, again, can lead to targeted vaccination, in this case with an expanded scope compared to what would be done based on clinical cases alone. The value of wastewater surveillance for the Global Polio Eradication Initiative is highlighted by the investments made by the Global Polio Eradication Program and WHO, and there are now thousands of sites where wastewater surveillance is conducted for poliovirus, often from informal sewage networks. So for typhoid, that use of informal sewage networks has been really key. So typhoid conjugate vaccines were pre-qualified and recommended by WHO, typhoid conjugate vaccines in 2018, but their uptake has been quite slow, and that's partly because of a lack of evidence on the clinical burden. There's very limited blood culture-based surveillance for typhoid. So the question here is, can we use wastewater detection of Salmonella typhi to motivate and monitor the introduction of typhoid conjugate vaccine? In this case, we're having to sample from informal sewage networks, drainage channels, rivers. So it's a very different system to what we would have in a high-income country. But it is possible, and what we're seeing here is the emergence of data to show that the prevalence of detection of Salmonella typhi in the environment does correlate with clinical incidence of disease. So can we learn from polio and typhoid and be ambitious about wastewater surveillance for other targets? And I think everyone here is enthusiastic about wastewater surveillance. That's why we're here. So one area that I wanted to highlight is the role of wastewater surveillance, not just in monitoring pandemics, but also in pandemic prevention. In other words, can we use wastewater surveillance to detect spillover of infection from their animal reservoirs? Particularly, can we use wastewater surveillance to estimate the extent of zoonotic infection and associated risk factors, and look for evidence of human transmission and genetic adaptation, potentially providing an early warning of threats? If we can do that, then we can implement risk mitigation activities, not only in public health, but across sectors, and also start to develop those medical countermeasures that we might need, including vaccines and diagnostics. So this is a focus of a new consortium that's funded by the Bill and Melinda Gates Foundation and the Hong Kong Jockey Club, working in Africa and Asia to develop methods and pilot those methods for detection of these infections in human wastewater. So finally, there are, of course, opportunities for wastewater surveillance. That's why we're here. But there are challenges. I don't have time to go through all these points, but I did want to raise a couple of those challenges. Firstly, there are technical challenges, of course. The methods for sample concentration and detection might vary between pathogens. So if we want to do surveillance, integrated surveillance for multiple pathogens, work needs to be done on developing appropriate methods to achieve that. And there's some real progress there and open access protocols for doing that. We also need to design appropriate systems that sample from the right places with the right frequency. And the kind of tools that we need to do that are emerging. So for example, if we want to choose and characterize sites, you can go to es.world and use their online tools to do that. So there are, again, resources emerging to help with that. In terms of costs and sustainability, I think we have a session this afternoon focused on that, and also a session on ethics, which I'm really looking forward to. And so just to end, I think the key point, though, is we can detect these pathogens, but we also need to generate data that's useful and used in public health. So thank you.
Thank you, Professor Grassi, for your informative presentation on the public health impact of wastewater surveillance for multiple pathogens. And myself, I deeply believe the multiple pathogens will be the future trend in wastewater-based epidemiology if the challenges you indicated are addressed. And our next speaker is the Alfonso Zecconi. He's a veterinarian and full professor of domestic animal infectious diseases with a research focus on the public health aspects of animal diseases. More recently, his focus has shifted to explore the scientific aspects of antimicrobial resistance through a One Health approach, and particularly in the epidemiology of antimicrobial resistance in wastewater in areas with large human populations and food producing animals. And today, the Professor Zecconi will present us the risk of antimicrobial resistance spread through wastewater in a One Health perspective. Okay, Professor, the floor is yours. Okay.
Thank you very much. Good morning. Thank you for this invitation. I'm probably only a few vets. I know there's at least another one in the room, so something different. So I think we can all agree on saying that antimicrobial resistance is one of the major, if not the major endemic disease that we have now affecting the global health. And on the other side, we can also agree on the fact that One Health approach probably is the most appropriate and most efficient way to cope with this problem. One Health approach now is included also in European legislation, in different documents, in different piece of legislation. The concept of One Health is written even in legislation. So at this point, my job is very easy because I have to convince you, if I need to do that, that wastewater-based surveillance, wastewater epidemiology fits very well in One Health approach to related to antimicrobial disease, but also to the pathogen, all the pathogen, viruses and bacteria pathogens. So wastewater-based surveillance fits perfectly in the interface between the three areas, so the human, the animal, and the environmental one. To show that with some more detail, I prepared this figure. So you see, it's more simple than it looks like. The red arrows are the potential connection, the potential way of transferring antimicrobial resistance, but also pathogens from the human side, the animal side, to the wastewater, and from the wastewater to the environment. So we can have through the water, to the drinking water, to the fertilization, irrigation, and so on. Then, thanks to feed and food, they come back to the human side. Of food come to the human side, feed come to the animal side. So closing in some way the cycle, even if could be a vicious cycle if we are not able to control this problem at the environmental level. So as you know, we have very short presentation. So I emphasize I think we need to think about. One is the problem that we are more frequently, at least in Europe, with a lot of situation where we have heavy rains. Heavy rains means that the wastewater plants are unable to process all the water coming into the plant. So this water with the spillways go directly to the water basins and directly to the environment without any treatment and without any control. So since this is a problem that is coming more and more frequently, at least in Europe, this will be another threat for the global health due to the presence on this water, both of pathogens and obviously resistant pathogens. The other point we have to think about that if we have no doubt that we can transfer pathogens, commensal bacteria, and resistant bacteria to the wastewater treatment plant and to the environment, we have to think about that when they come out, due to the recombination of these bacteria, we are increasing the pool of pathogens with bacteria with antimicrobial resistance. And also we can have changes in the pathogen and the virus factors of these bacteria. So when we talk about wastewater-based surveillance and epidemiology, we have to think about to check in the way in and the way out to have a more precise figure of what is going on. How to do this? We already have some of this suggestion, and probably currently the best way to do is to work with the next generation sequencing that in the past was quite expensive. You need to have a very sophisticated labs. Now there are tools that can be applied in a less sophisticated, we can say, the labs, and also they can then be moved among labs. And one of these technologies is a nanopod technology. I think the Professor Grossley showed a piece of that in his slide, and we probably will listen more to this technology in the future presentation. So this is something that could be applied even if you are not in the best place from the molecular biology point of view. So we can get a lot of information, so we can define the resistome in the case of wastewater, but also obviously in other situations. To put the theory in practice, these are only some of the things we, not only myself, but most of the colleagues are behind my presentation that we show the names in the next one. We are a part of the 4E Plus Alliance, so we are working on this way. These are some of the potential, or some that we did, topics that we can we perform in the past, and we are trying to do in the future in the area of antimicrobial resistance and also to the surveillance of wastewater. So these are the people in the group. They are people that really drive the research and they work in this on this topic from our University of Warsaw, from the IBMC from Strasbourg, and obviously my University in Milan. Thank you.
Okay. Thank you, Professor Zecconi, for your insightful remarks on monitoring antimicrobial resistance. I think the wastewater surveillance is emerging as an effective monitoring tool for detection of AMR and AMG, which is called as a silent pandemic by the World Health Organization. And as you all know, with the recently published revised European Union Wastewater Directive, the European Union member state countries will be mandated for the monitoring of the AMR in the wastewater treatment plant serving more than 100,000 population equivalent, which will be a milestone for the AMR detection in wastewater, I think. And our last speaker is Laila Toro. She's an epidemiologist at the French National Public Health Agency. She is part of the SUMO team focusing on the routine microbiological surveillance on wastewater nationwide. With this role, she contributes to various projects in the field, including developing a wastewater surveillance strategy for the Olympic and Paralympic Games. She participates in the European Union Wastewater Integrated Surveillance for Public Health EU-WISH joint action aimed at enhancing wastewater surveillance for public health across Europe. And today, Laila will present us the wastewater surveillance for the Paris 2024 Olympic Games. Yes, Laila, the floor is yours.
Thank you, Bilge, for that lovely introduction. And thank you to the organizers for giving me the opportunity to present. I'm very pleased to be here. Today, I'm going to present a brief overview of the experimental wastewater surveillance system that we implemented for the Paris 2024 Olympic and Paralympic Games. But first, a little bit of context. So the World Health Organization characterizes a mass gathering as an event where the number of people attending is sufficient to strain the planning and response resources of the community, state, or nation hosting the event. Such gatherings have inherent characteristics that place them at a greater risk of infectious disease transmission. Notably, they involve concentrations of large amounts of people at the same place at the same time, which can facilitate pathogen dissemination. And they can also see a considerable influx of international travelers, which can increase the risk of disease importation. So there's been a growing interest in applying wastewater surveillance to these kinds of settings, though currently few concrete examples exist in the literature. With the Olympic and Paralympic Games being hosted by France in 2024, we saw a unique opportunity to implement an exploratory wastewater surveillance system tailored to the Games. Our objectives for this project were first to gain experience in applying wastewater surveillance to mass gatherings, as it's something that we hadn't done previously, and to explore whether wastewater surveillance can provide added value to other population-based surveillance systems during a mass gathering event like the Olympic and Paralympic Games. To identify priority pathogen targets that are both relevant and feasible to monitor in wastewater for such an event, we started by drafting a preliminary list of pathogens of general interest for public health surveillance during a mass gathering. And then these were evaluated against 3 inclusion criteria: A, analytical feasibility; B, relevance with regards to the specificities of the event and characteristics of the pathogen; and C, added value to inform public health decision-making. And so this allowed us to identify 6 priority pathogen targets that are suitable for wastewater surveillance during the 2024 Olympic and Paralympic Games, or OPG. These were poliovirus, mpox virus, influenza A virus, influenza B virus, SARS-CoV-2, and measles virus. If you'd like a more detailed account of this portion of the project, we invite you to consult an article that we recently had published in Eurosurveillance that covers this in more detail. Then, to define a wastewater surveillance strategy for the Games, we looked at the possibility of sampling at different kinds of sites. So we not only looked at the possibility of sampling at the entrance of different wastewater treatment plants, but also at the possibility of sampling specific points on the Parisian wastewater network that would receive the effluents either of specific buildings of interest, such as sports venues, cultural sites, points of entry and means of transport, as well as other subsections of the wastewater network. This allowed us to identify 10 sampling points of interest, though due to technical and logistical constraints, the wastewater surveillance system implemented monitored the 6 pathogens identified across 5 of these sampling sites. These were Saint-Centre wastewater treatment plant that was sampled biweekly, as well as 4 points on the Parisian wastewater network that you can see here to your left that correspond to 4 catchment areas of the city. These were sampled weekly. Of note, for sampling on the wastewater network, we were able to capitalize on work being done as part of a thesis project by Morgane Levert. And in total, the surveillance for the Olympics took place over a 16-week period. All primary molecular analyses and pathogen screening was done by colleagues at OBEPINE. And then for samples that would be positive for poliovirus, mpox virus, or measles virus, diagnostic confirmation was assured by their corresponding national reference centers. We also had follow-up meetings organized by the French General Directorate for Health. These had representatives from Santé Publique France, Haute-Paris, as well as the different national reference centers and laboratories for the 6 pathogens monitored. And so the purpose of these meetings was to discuss results, their interpretation, and the need for further action. So we're still in the process of consolidating data, but I do have some preliminary results to share with you today. So here we can see for each of the 6 pathogens monitored, the proportion of sampling locations across different categories of viral circulation. For SARS-CoV-2 on the bottom left, we see that we had more or less continuous signals, and this was consistent with clinical data. For influenza A, we also had more or less continuous signals, but that started a little bit after the beginning of the monitoring period. And this was an interesting signal for France in this period of the year. For influenza B, we had a single detection the last week of monitoring. For mpox virus, we had 1 positive sample confirmed by the National Reference Center. And this was consistent with clinical data. So there was a clinical case reported around the same period. For measles virus, there were multiple detections throughout, but analytical methods for diagnostic confirmation are still being validated by the National Reference Center. So we don't have too much more to say about these for the time being. And for poliovirus, we saw no detections. So as takeaway messages, we wanted to once again highlight the importance and the value of an interdisciplinary multi-partner approach for a project of this nature that was essential for us both in the planning portion and the implementation portion of this project. And to conclude, we hope that our experience can serve as a starting point for others who might be interested in applying wastewater surveillance to mass gatherings, as we feel that it's a tool that has a great deal of potential in this context. And then a special thank you once again to all of our partners without whom this project would not have been possible. And thank you all for listening.
Okay, thank you, Laila, for presenting us the strategy followed and takeaway messages learned from the application of wastewater surveillance in Paris Olympic Games. I deeply congratulate you and your team for this best practice, demonstrating the power of using wastewater surveillance for quick scanning of the public under such big events, okay? And now we have time, yes, 25 minutes for the QA session. Okay, first I would like to start myself by directing question to Vincent, and then I will take questions from the room, and if you have any question from the virtual connection, I will also take this. We have enough time, I think. Yes, Vincent, first I would like to ask you, how did you choose the wastewater treatment plants? Which criteria you followed while they are deciding the treatment plants?
Yeah, thank you for the question. It's a very important question. Actually, we probably will need to talk about sampling in wastewaters and outside of wastewater treatment plants. So basically, this work has been done by Yvo Madej, who is here, and his team. We basically use 40 different criteria, including demographic criteria, economical activity, transportations. We need to have a look on what happens during vacations, for example. If it's not so easy to get mobility data, you need to make sure to monitor this kind of places. So we use more or less 40 different criteria to choose them. And just to remind you, we have 22,000 wastewater treatment plants in France, and we have to sample 200 of them.
So it shows the logistics, setting up the correct logistics is behind the wastewater surveillance activities is quite important. I think it is the letter A to start nationwide wastewater surveillance activities.
It's a very important point.
Okay,
really.
thank you. Now we can take the question from the room. Can you raise your hand, and then the microphone will be directed to the audience if you have a question. Okay, while we are, okay, yes, please, okay. Can you introduce yourself very briefly, your name and your organization, and then please direct the question.
Yep.
Thank you. Thank you very much. Émilie Perron from WHO. I'm epidemiologist in the Surveillance Systems Department. My question is for Leila. What kind of actions were actually taken after the detection of any signals from wastewater during the Olympics and Paralympics in Paris?
Thank you for the question. I think we wanted to be, since it was the first experience in applying wastewater surveillance for France with mass gatherings, we wanted to be very prudent in how we interpreted wastewater signals. So we didn't feel that any of the signals that were produced warranted any specific public health action.
Okay, thank you, Naila. Maybe we can direct the online questions. Diane, how we will direct the questions coming from the, I couldn't see the questions. Yes, please.
Okay, can you hear me? Yeah.
Yes.
Perhaps I can begin with one general question from the chat. Is it possible to share the slide to all the participants? Yes. The slides. The participant asks the slides to be shared after the session. Is it possible or not? Yes, okay. So the second question perhaps is for Vincent. The question was, has OBEPINE assessed the surveillance of antibiotic microorganism resistance also?
No, actually we didn't, just because in France we found a lot of work that has been done on anymore wastewater, so we don't want to compete between networks, but really this is something quite important, and we try now to move to this question and try to federate people working on this question. So we didn't do it, but we clearly want to do it.
Okay, another question, if I...
Yes, sure.
Yes. There are some questions asking for the best method to evaluate antibiotic microbial resistance in wastewater. Should it be only sequencing or only targeted antibiotic resistant genes, phenotyping, or a combination of such methods? And other question just ask that metagenomics methods are not enough in our environmental surveillance until we make isolate.
Okay, Alfonso, I think you can answer this question very easily.
Thank you. Thank you for the, it's a quite good question. So thanks for the question, because then I can occupy the 25 minutes that we have for questions, trying to answer to that. In short, I think that the sequencing up to now probably is not the perfect way, but is the most efficient one particularly because we also have the tools that allow to check it in different conditions, in different general and economic conditions. So obviously we can do many other things, but could be more expensive, more time consuming. So if you want to be effective, to be on time, probably the sequencing that we call the resistome approach is probably the most efficient. And not to say the best one in absolute, but is the most efficient. I personally am not in favor to look for specific resistant genes, so to go directly to them. And I think that now to talk about phenotypes is a little too old, if I can say in this way. We need to go to something that is much more related to molecular biology.
Alfonso, I can ask one more question.
Okay.
I'm giving extra time to you, okay? So, okay, you will forgive me, okay? As an environmental engineer myself, I would like to ask, I know the answer, but I would like to ask you for our audience. What do you think about the efficiency of the existing wastewater treatment plants around the world to decrease the AMR and AMG? Are they able to treat them?
So this I'm like to be a student with. Honestly, I don't think so. I mean,
Yes.
particularly, I mean, probably we can be some way more safe concerning the pathogens. But if you look at the commensals could transfer a lot of antimicrobial resistant genes. And then normally they are not controlled by any kind of treatment. So the risk to have this kind of bacteria in the environment, and then also the changes, the modification, the transferring of the resistant genes in the environment, keep the risk to getting them back to the human and animal side is not negligible. So I think the real problem, I say for the previous question, we need to look at the resistome in a different steps in the environment.
And we can give message to environmental engineers. They have to focus on the new technologies, hybrid technologies for the removal of the AM, not only the AMB, I mean, not the bacteria, but also for the genes.
Yes, yes,
okay.
yes.
Okay, thank you. Thank you. Is there any other question? Yes.
In the chat, yeah, we have some. No.
There is one from the room. Yes, please. Vincent, this side. Oh, okay. Okay, next we can take this. Okay, okay.
Thank you for your very good presentation. And I also have some question. Magdalena Popowska from University of Warsaw, because I am working with AMR in the environment. And for me, it's very important One Health approach in this content. That's, I think, your question. I don't remember who was. It's just the question like, what was the first, eggs or chicken? Because for the microbiologists and biotechnologists, it's much more important to microbiome, resistome, plasmidome. Yes, because the mobile genetic elements and everything is so very, very important. That's the big question is how much money we have to do this, the first question, and then World Health Organization have some recommendation for European countries, and we should working on the strategy to control, monitoring, control, and prevention of AMR in each of sector, One Health sector. That's it's difficult to answer, I know, but I think that's also good. I don't know how is your opinion about qPCR cheap strategies to just monitoring antibiotic resistance genes, and maybe some how to say molecular vector of dissemination of AMR. I mean E. coli, because it's found everywhere.
I said before that I need 25 minutes, so I can say, I mean, I agree in what you say. Again, it's difficult to give a single best solution because it's quite a complicated argument. It's quite complicated. The environment is quite complicated with so many different facets, so it's not so easy. It's important to consider. So once more, when we talk about One Health is relatively new, but it's not really new. but we still need to think about in a within a 1F approach. That means we have to consider, if you are on a physician or a human side, the animal and environmental. For people working with animals, is a little bit easier to think about the environmental part, but still not enough. So we need to consider all of them. Then we may develop a strategy, a common strategy. Could be easier, at least in Europe, could be easier in other countries, but we need to talk about that. We need to work on that and step by step to try to define the best option. We will never find the best one, but we need to work on this to find what is feasible, because then the point is we have talk about money. We need to have money to that, but it's not so critical. So we now have technology that could help to have this information without spending too much money. So, and we have to discuss that. This is quite important, and I'm quite pleased that I raised so many questions in this conference, because this is important to talk about that, and we need to work in this side, and involving the environmental engineer, all the people working in the environment, because up to now, in many cases, there is no real connection. Mm-hmm. So this is also important to develop this kind of connection with this kind of areas.
Okay, thank you. Yes, please.
Good morning. I'm Lourdes Oliveira from Oswaldo Cruz Foundation, Minister of Health, Brazil. And so thanks so much for your presentations. In fact, I have two questions. The first one is to Laila about preparedness. So in case of positivity, which was your plan to respond? I mean, measles in Americas, for example, it's eliminated. Polio, the same. So in case of positive, you must be very fast to avoid the transmission. And for diseases, you have a high transmissibility as measles. So what was your plan in the sense? Because of course, firstly, you have wastewater, it's a signal, it's not a clinical diagnostics. And so how are you working on that? Because it's extremely important. I think your experience to other countries which are going to receive mass gathering. The second question is open to all of you, because all of you are working with that. For example, for poliovirus as bacteria or as fungi, there's a part of specialists which defend that we must make isolation. If we do that, we are losing the opportunity and the big advantage of wastewater, that it's early warning. And so I would like to listen you in your experience to discuss about these positions. We can make direct detection. This is very, we have the opportunity against, no, we really do need to make a viral isolation or bacteria or fungi isolation to afterwards have any kind of action. So I would like very much to listen to you. Thank you very much.
Okay. Yes, Laila.
Thank you for the question. I think, again, of note, the wastewater surveillance system implemented for the Olympic and Paralympic Games was an exploratory project, a first experience. So once again, we wanted to be very prudent in interpreting wastewater surveillance systems. And as I mentioned, diagnostic confirmation is still being, methods are still being confirmed by the National Reference Center. So I think that the way that we envisioned dealing with this was through our weekly meetings with different experts, but we were not able to put into place any specific action for these detections. What else? I hope that answers your question.
Okay, maybe we can pass to the second question. Nick, can you answer the second question? Isolation of the viruses.
Yeah, thank you, Bouquet. Okay, so I'm not a microbiologist, but I do agree with Alfonso that sequencing is really helpful here. So we have seen some problems with qPCR being non-specific in wastewater because of both genes, horizontal gene transfer in bacteria in particular, and off-target amplifications. So when we have a highly abundant pathogen in wastewater, I think qPCR or digital PCR is fine. But when we're looking at very rare detections, I don't believe that those methods are any longer reliable because of that issue of non-specificity. So we do need some kind of confirmation that could be through isolation or it could be through sequencing. I think sequencing has a lot of power and it's a lot more high throughput. And for many of the pathogens we're interested in, confirmation can be made based on a sequence alone. But it may be more challenging, I guess, for some bacteria where we have a lot of horizontal gene transfer, and you may feel more comfortable if you're able to isolate that bacterium.
Okay, thank you. Yes, please, your question. Okay, yes, your turn. Yes, yes, please, your turn.
Okay, hi everyone. My name is Katrina. I'm a director at the data analytics business in London called Affinity. And see, I picked up on this in Nicola's presentation around challenges, and I was quite surprised to see that timely and effective data sharing and standardization wasn't on there. And we all know we're stifling innovation and progress by not making raw data and adequate metadata available. And that's not like a fancy heat map on a website, I would say. And I wanted to hear what your perception is of this challenge. And is this a lack of knowledge, what adequate data looks like and what, you know, best practices look like, or is this protectionism?
Okay, shall I take that question, Bouguet? Yeah. Thank you for that question. I didn't put all the challenges. That's definitely one that's foremost in my mind, along with the ethical side as well. I think there are two aspects. One is what is recorded. So there need to be some kind of data standards. And I know there are a number of projects underway to create those data standards. So I think the PHAGE project, for example, is one of those that are developing those standards. I think WHO has a key role to play in encouraging the use of those data standards. The second part is about what you alluded to in terms of sharing data and reporting data, both within countries and between countries. And there, obviously, there can be challenges. There are ethical aspects. There are also risks of negative consequences, for example, reporting a new SARS-CoV-2 variant. So it would be sort of in the context of more generally reporting pathogen genomic data, great to see some kind of encouragement of ways of reporting that data on platforms. But it's certainly hard, and I don't see any easy solutions.
Okay, yes, please.
Yes, hello. My name is Arnaud Tarantola, and I'm a colleague of Leila and Damien. I'm a medical doctor, epidemiologist, and I'm the head of the epidemiology field bureau for Île-de-France, for Paris and Greater Paris area. And our team was the first line response team and investigation team during the Paris Olympics and Paralympics. And in response to our colleague's question about what was done with the data, and Leila agrees with this, I'm sure. For one thing, the team at the water investigations unit basically would launch complementary investigations, for example, the genotype of the Mpox virus that was isolated, et cetera, et cetera. The second thing is that our team was responsible for working with a regional health authority to do investigations should they be warranted. The pathogens that Leila showed were basically endemic except for poliovirus. We had measles, we knew that. We had influenza. This was not the right time of the year to have influenza, but we did detect some cases. It had been anticipated in a risk analysis that had been done for the Olympics, trying to identify the sort of pathogens that could be imported and the risks that would emerge. So we had anticipated that influenza would be mass imported from the Southern Hemisphere at that time, but not in such a way that we needed to implement a mass vaccination campaign. As for the other pathogens, Mpox we had, measles we had. The one thing I would like to stress is that we did exchange the information. And one of the things we did is that we were talking each day with our bulletins to the emergency room physicians, because it's a rather well-known fact in surveillance that influenza cases, for example, rise in the data as soon as we tell the clinicians that it's the start of the influenza season. Gastroenteritis rises as soon as we tell them that there's gastroenteritis, and bronchiolitis rises, etc. What I'm saying is that the early warning is not just to the authorities, but to the clinicians, because they increase the sensitivity of their capacity to detect and name these pathologies in their patients. And then we see stuff appearing in the data. And this may be one of the reasons why there is early warning, not because they don't see patients, but because they call them influenza patients. Thank you.
Thank you. Okay, then let's close the session. Okay, be on time. Okay, as we conclude this insightful session on wastewater-based epidemiology for pathogen monitoring, I would like to extend first my gratitude to all our distinguished speakers for their valuable contributions and in-depth presentations. I would also like to thank our in-person attending and virtually connecting audience for their active participation and their questions. Certainly, I would like to highlight that our conference will continue with a series of compelling sessions. We will delve into data analytics just after this one, explore diverse sanitation infrastructures, discuss ethical and legal considerations, and gain insights from global perspectives. Thank you once again for your participation. Okay, I look forward to listening to the upcoming sessions. Have a good listening. Okay, thank you.
It's time now for our second topic, wastewater surveillance for public health. And the chair of this session is Yvon Madé from Sorbonne University. Welcome, Yvon.
Okay, so good morning. I'm Yvon Madé. I'm professor of applied, I would say, interdisciplinary mathematics at Sorbonne University. And I have the pleasure to introduce Anna Maria de Roda-Hutzman. So she's head of the environmental department at the RIVM, which is the Dutch National Institute for Public Health and Environment. And she's also a shareholder in global changes and environmentally transmitted infectious disease at Utrecht University. So she's a specialist of molecular biology, water virologist, and she's interested in both surveillance of air and water, what we breathe, what we drink. And she has been also instrumental in the Dutch detection of a virus. She's a wonderful speaker, and I'm very pleased to give her the chair now.
Thank you so much. Thank you so much. Well, after this splendid introduction of Yvon, I always have this very terrible moment of having to lower the microphone because I'm so short. So again, my warmest gratitude to Yvon and Diane for inviting me to this wonderful conference of the OBEPINE network. I've been listening with great interest to the first session, which was already great discussions, very apt questions. I hope I can tie all of the things that have been raised before in the session and also tie in some of the later points that will be discussed in one presentation, but not keep you from your coffee. So with that, I would like to start with a little bit of discussion about the relevance of our terminology. Because I don't know if you listened carefully this morning already and also looked at the program, but we seem to be able to be very apt at messing up our terminology. And this may seem trivial to all of us guys, like being more in research, but I know that like my other half in public health, I know that this has been a real challenge for the people in epidemiology, but also some of the policymakers. What are we talking about? So what I did on this slide, I put a lot of text on the right-hand side, but that's the WHO definitions that's on their website. So I'm not going to read out that text, but I find it very helpful to make a distinction. But I wanted to point out the figure on the left, because this is what we always use to sort of tease out what we are talking about. So on the most left-hand side, we're talking about raw sewage, so domestic waste, domestic wastewater, waste that comes from people. And what we can do with that is look back up the human body, so to speak, literally, and see what is circulating in the population, in the human population. Of course, one of the first comments that we will get from the clinicians and some of the public health professionals is, but what about the pigeons? What about the rats? I mean, they also, their feces also ends up in the wastewater. Well, this is some challenge I would like to put forward in this research community. We need to solve this. We need to be able to answer this very simple question, because this is all about trust in our data. The next thing we can do is, was also mentioned before in this morning session, is about what happens in the sewage treatment processes, especially with AMR, antimicrobial resistance. This has been addressed. And also, what's the efficiency of the treatment processes? So can we reduce the pathogens? Can we reduce the resistant bacteria? Can we reduce the resistant pathogens? That's a different field. When we move to the right, this is more about the environmental transmission routes. So what are we talking about? We're talking about combined sewer overflows. That's in our part of the world, the high income settings. But of course, the waste also ends up in the rivers and in other surface waters, and may even end up in the groundwaters, and can cause infections downstream of those discharge points. So just to make that clear, I think that's something we need to solve, because otherwise this will not help our funding. And then I would like to propose, this is very new also for me. I thought maybe we should see this much more as a continuous cycle, and not look at our data, not look at our scopes going left to right, and then it will end in nowhere. Because what do we do with our data? We put them on dashboards. Does something happen then? Probably not. Maybe, but we need those very accurate, like who presented that, Laila, on the Olympic Games? I love the way you explained, like, oh, we sit together every day or every week and discussed what we found. And I think this is very helpful. So there it should start. So we may have a societal challenge. It could be Olympic Games, but well, I think more generally around the globe, this will be the emergence of pathogens that we can then pick up in our waste, and make sure that specifically in those zones where our human surveillance, so the knowing what's circulating in the population is very low. I think our waste, those are hotspots usually for spillover from the animal reservoir, like with MERS coronaviruses. And I think this should be to our attention, and that's where I think wastewater surveillance can play a pivotal role. So this framework for environmental surveillance, to me, it sort of starts and ends and keeps going all around these societal challenges, and I will explain a bit more about it later. I think from the challenges, it actually goes to thinking about ownership. So who is actually responsible? Who's the authority that should actually put these programs into place? Who should look into what comes out of this environmental surveillance for public health and communicate about it? So I think what we know best is actually the design and the implementation and the data analysis and the outcomes. But I think we should pay a bit more attention to the communication, but also to the public health action, and then come back to the societal challenges. So what has it brought us? And again, I thought a very honest answer about the study on the Olympic Games is like, okay, so this was a kind of a first, and we need to learn from this experience. And we need to move ahead and see whether we can make it much more actionable. So with respect to the societal challenges, I thought it would be a good idea to put those into relations with the Sustainable Development Goals. I think they're seem to be a bit out of fashion. I didn't hear a lot about them anymore, but I would like to take the stage and put them at the front. And I think this is a very nice way that it was posed, how you can translate the Sustainable Development Goals, for instance, goal number 8 on decent work and economic growth, into create job opportunities for youth. So what was done here was translate these to actionable goals. And when, surprising to me, when I thought about this, I thought actually there's a lot of these goals that relate to wastewater surveillance and what it can bring as a surveillance tool. A nice study by our colleague Rolf Halden has actually translated the goals. Take a look at that paper if you haven't seen it yet. Has looked into all these sustainable development goals, and well, I would say no poverty can also relate, but I just followed their example, and so they ticked all the boxes and said, look, all of these the wastewater surveillance can help with. And today we're together in a great partnership. So I guess we can take on that last one as well, goal number 17. So that is a bit about the societal challenges and how they can relate to wastewater surveillance and benefit. One example that I got from our colleagues, and I don't dare pronounce it, but it was a nice study in Stoughton, about wastewater-based epidemiology as tool for hazardous materials such as metals. And what they said is actually human biomonitoring for these metals is insufficient, but wastewater surveillance can help us. So I thought it was an outstanding example where actually there is a need, there is a question, there's a societal challenge, there can be ownership, and then we can relate and then do all the things that we like to do, is do the analysis, do the analysis of the wastewater, do the analysis of the data, and then come up with an outcome. So very nice example. With respect to ownership, I would like to stress that the objectives and scope of the wastewater surveillance are really important. So I guess when you set up a study, set up a program, the first thing you need to do is really sit down and think about why is this important to do these studies? Why is this particular study, why is it important? Because if the answer would be that this is just about doing nice research, then I would like to ask you to think again, because I think wastewater surveillance for public health can have consequences. I remember last year being at a very nice conference, actually the predecessor of a conference that we are now organizing in the Netherlands, where someone raised this point of, yes, I'm so excited, we found poliovirus in wastewater. I was going like, what? Did you? Yes, it's wonderful. I will have a very nice publication that's going, yes, but this has consequences. We try to eradicate polio from the world. So don't think that your research that the only meaning it has is to come up with a nice publication. So it scares the hell out of me. And then identify your stakeholders, assemble the team. We know this from other frameworks, like for water safety planning and sanitation safety planning, which are quite closely related to our wastewater surveillance. It's important to sit down, to attract both from the public health side and the environmental side, for instance, on sanitation, attract those experts and discuss together what is the objective, but also what can be done with the outcomes. And also, I think it's important to attract some of the other people, other partners that may be involved or interested, like community representatives are also very important. They may be in charge of access to safe sanitation, or and it can also be very, very relevant to them. And of course, people like Bilge, the engineers, we shouldn't forget. And then the scientists, but I imagine we don't forget the scientists. One very nice example of our colleagues, Anansi, Taru, Tarja, maybe she's online, I don't know, was this stakeholder survey. And actually, to me, this was the first example where I thought, oh, this was properly done. So in 3 African countries, there were workshops and surveys about how to embark on a wastewater surveillance program, how to deal with infectious diseases and antimicrobial resistance, how to prioritize pathogens in the program, and then do all of this before starting the research. So an excellent example of how this can be done. When we look into design, I noticed how the baseline is important. So a baseline can be, okay, we don't have a sewer setting. So we'll hear in the next session, I think, very nice panel to discuss non-sewer settings and wastewater surveillance or waste surveillance. And in the world, this is the case in most member states. So that's important. On the other hand, under the Protocol on Water and Health, we have learned that with member states under that protocol, the starting point may be quite different. So these may not be scientists in the environmental domain at universities or knowledge institutes, but these may be clinical settings. So it may be that they do have a PCR apparatus, they have sequencing, but their baseline is quite different. They know nothing about wastewater or waste, or not a lot. Sometimes when you talk to the hospital hygienist, of course, they may know a bit about the sewer lines in the hospitals, but it's a very different starting point. So remember with your design to know the starting point. What is already known, what's already in place, what do you need? A strategic sampling approach is put here very simply, but this can also be an enormous challenge. Where to sample, how much to sample, how to transport. But I think we hear more about it today. And what I wanted to focus a little bit on was also whether there's any legislative or other framework that requires us to do this sampling and maybe determines part of how we design the sampling or the sewage surveillance program. The safety of personnel and I think ethical aspects we will learn about more this afternoon, but to show you a bit more about those legislative frameworks. One of them, of course, is the Global Health Security Agenda. It's a very different domain from most of the domains that you may be working in, but I think it's a very important basis. And this GHSA is trying to do a lot to bring people together, for instance, in terms of antimicrobial resistance. And we also, of course, have the WHO Global Action Plan on antimicrobial resistance, which also addresses wastewater surveillance. And I noticed how much these fields are siloed. So it's very difficult to bring everybody together. Then, of course, we heard from Bilge that this European directive on urban wastewater treatment has been recasted. And we were very fortunate to learn a couple of weeks ago that at least antimicrobial resistance is at the heart of this directive that will actually spur us on to look into this. The protocol on water and health I just mentioned, so we'll not go into that, but also in your national strategies, there may be attention to wastewater surveillance. So in the Netherlands, we have this Dutch global health strategy where we want to take care of global health together. I was very happy to hear that here at Sorbonne University, there's also a global health partnership, I understood. So that was really interesting. Then to the implementation phase. I think one of the things we would have liked to have before the pandemic broke out was wastewater samples that we already stored. So when you think about the infrastructure, think about the storage. And I know in the Netherlands, and I'm very happy to have my colleague here, knows all about these hundreds, thousands, tens of thousands of samples that are already stored in the Netherlands. But only very project-based samples were stored before the pandemic broke out. So we did have like a year like 2016 that we had some samples and some others, but not in a systematic way. But it's very difficult in times when pandemics are slowing down to keep that up because it's very expensive. So I would like to challenge you and think about a very cheap, simple way to storage samples to be able to go back to when something happens. You need to think about, of course, the disposables, consumables. We need to have protocols, pipelines, personnel should be trained. This is not a small task to achieve. But our colleagues in, for instance, well, all around the world, but one example of our colleagues in Alberta, in Canada, they thought about everything that's needed in a very nice review that they did, where they talk about general sampling considerations, sample sources and locations, all of the above, and also about data sharing and reporting and transparency. What I would like to stress is what's quite interesting is that with SARS-CoV-2, for the very first time, because we had been doing, well, I'm kind of old. So what we have been doing in wastewater surveillance for a very long time already was look at noroviruses, look at all kinds of viruses, polioviruses, but also bacteria, AMR, before COVID. But this high level of quantification, that's just amazing how we can follow these trends and how we can already see that we will have hospitalization figures go up. That's very, very special that we saw that with COVID. One of our most loyal users in the Netherlands was a clinical doctor in one of the smaller towns who said, I don't look at anything else. I just observe the wastewater surveillance data because they tell me everything. So, of course, these are the people that you want on your social media. With respect to outcomes, what I would like to stress mostly is we saw a lot of these interesting graphs and figures. I was much impressed by our French colleagues and their data, and my colleagues in the Netherlands can also show very fancy graphs and having a lot of samples, having a lot of sequence data. But I think defining the data needs is a very important point. So do we actually need sequencing? What do we need to answer the question? What do we need to comply with the legislation? It's very important to take that again as the starting point for what you do. And then on the side, do some more research. But let's see how much is needed for our objectives. What I also would like to stress is that in many parts of the world, most parts of the world, we don't have all these tools and all the funding available. You see much more coming up, so that's really good. But I think we shouldn't neglect very simple tools. So one of the challenges would be for me, like, isn't there a very simple way to detect pathogens in the wastewater, just sticking in something and then having a signal like what we had with the positive test for SARS-CoV-2? But then not putting it up your nose, I mean, that's okay. What's also a very important point is the data cleaning. So we know that if you have a lot of data, it's very important to go through all those steps. So what's going on with the data? What am I seeing? You need all these controls, normalization, how to interpret. And again, this is very important for the trust in the data and the validity of it. And there's a very nice example of colleagues of ours in the Netherlands that did a very thorough study on one of our airports, Schiphol Airport, that you may know, although maybe you pronounce it differently. So what I also would like to point out is that important in this respect is that variant risk assessment is actually quite important in the clinical domain. And I noticed how actually there's again some divide between how this variant risk assessment is approached. We are very fortunate to be in this new COFINET network of the WHO headquarters with our colleague Lorenzo and his team, where they have all the clinical labs that take care of looking at the first signals of new coronaviruses, both SARS, MERS, or SARS-CoV-2. And they then look at the clinical severity, the growth advantage, the antibody escape, and then determine the level of risk that that variant poses to the human population. This, I think, should be much closer to what we are trying to do in the wastewater surveillance. And fortunately, there are 4 environmental laboratories, one from all over the world, who are now involved in this human coronavirus network. So you'll hear more about that from us. One of the other examples I would like to give is on antimicrobial resistance. We had a large program with baseline measurements in 2015, 2016, and we had some really neat outcomes. We saw, for instance, that the sensitivity of detecting resistant bacteria in the wastewater is very high. So it's a very good way to look at AMR in wastewater. And I'm very happy that Frank is also in the room. We worked together already some time ago on a global study on AMR. And we found that global sewage surveillance can also be useful even with sort of one-off sampling in data-limited regions. Improving sanitation and health can potentially limit the global AMR burden. That was one of the outcomes. So this is very, very encouraging. And in the Netherlands, we're now continuing with our AMR program, and we're actually looking into new and emerging antibiotic-resistant bacteria, like the colistin-resistant. So over to communication. I was happy to see that WHO has a very neat website where they put all the dashboards together. So I didn't have to get all those figures and all these nice outcomes that you all have from all of your own countries, but they have actually collected all the global and local dashboards on their website. I don't know how up to date it is, but it's very neat, so check it out. What I would like to stress is that putting data on a dashboard doesn't mean that it's used for anything. So people are looking at it, but is that sufficient? That's what I wonder. Or should we do more? And again, I was very encouraged about these multidisciplinary groups. I think we need to work together over these silos and not see each other as competitors for funds, but work together and try and get this going. And I think there's a big difference between passive versus active communication. So I do have an example for that as well. Remember your target audience. So if you put stuff on a dashboard it doesn't mean that it's understandable for everyone. It means it's understandable for those people that were originally in the top of the heads of the people designing the dashboard. So remember that. And also people will have more attention for the websites or the information sources that they normally go to. So best to integrate it into their sources and what they always look at, and then see if we get more active participation. Public trust I already mentioned, but also I think we need to adhere with privacy legislation, but I think that also will be addressed this afternoon. So communication at a local level, this is the work that was mostly done by Annemere van der Drift in our center, in our institute, together with Anna Welling and the Municipal Health Services. And what I find very interesting in this roadmap for municipal health service professionals is that not only is it in one of their guidelines now to look at this data and how to act, but I would like to point out, and I don't know, I do, probably I mix it up and press the wrong button. Let me try. So here, what you see is when the number of cases, in this case, COVID cases, is down, you actually need more intensive sampling to be prepared for the rise. This is so difficult to communicate to funders. So nothing's going on in the world, and now we should have to monitor intensively, like ideally every day. But, and so what you propose is that if the pandemic is on the rise and it's growing and it's going on, it's all over the world, we don't actually need to monitor. Well, because it's already ongoing. So let's put our resources at that instance at what we can do about it to prevent further spread. And, you know, it's good to monitor, but especially in these times, these low times, or what do you call it, cold phase, then we should monitor intensively. But, well, that's difficult. So if you can help me and give me some arguments how to stress that to the policymakers, that would be very helpful. So one other example of one example of a public health action, which I would like to, no, sorry. In general, public health action is important with wastewater surveillance. If there's no action, if you don't have an action perspective, then, you know, think about it again, how you want to spend that budget. And one of the most important things here is the proactive governance. So during COVID times, I had ministers from all around the world phone me and say, there's these colleagues of yours, these researchers that have been giving me results, outcomes of the wastewater, and they're showing me stuff. And now I don't know what to do with them. And I had to redirect them to their own groups, because you need to decide beforehand. If I just hypothesize, if I find one of these pathogens, if I find 2, if I find 100, if I find the first variant, the second variant, or a third variant, who is then responsible? Who is going to communicate? Who is going to run this action perspective? Who is going to do what? Which public health action? And I know I'm preaching to people that already embrace this, but I think it's such an important point. So you need to have that clarified before you start. And then there's diversity of public health actions. I don't have to tell you about it, because we had some really nice examples, from awareness raising all the way to targeted vaccination campaigns. Although I'm always very curious, I don't know where you are here, Nicholas, but what was the uptake of the vaccinations for polio? Was there a good response? Well, that's, yeah, sorry. Yeah. Sorry to blow the balloon, but yeah, thank you. And also public health action, of course, you need to think about the local all the way to the global level. What can you do with the data? One very neat example, sorry, that's what I started with the previous slide, but my dear colleague in Brazil, her group, Marise, has had a very nice local public health actionability in a small part of Brazil, where actually in the first stages of COVID hitting Brazil, they could use the wastewater surveillance data to help families, for instance, that didn't have the funds to isolate themselves. They provided funding for these families to go to hotels and self-isolate. And I thought, well, isn't this one of the most beautiful examples of a public health action that you've heard, because it was all based on the wastewater results. So coming to the end of the presentation, one other example, I think, is one that you may find interesting. I heard a lot about polio already. This is about a publication in Eurosurveillance about biosafety. So we all now know that we're coming close to polio eradication globally. We need to keep the virus safe. So we need to have facilities that are able to contain the poliovirus, make sure it doesn't end up in the environment. And what happened in our facility producing poliovirus vaccines, there was someone who got actually infected, and by doing wastewater surveillance, we could pick up the signal, isolate the person, and look into it a bit more to see whether this was not causing any major problems, just as a good example. So what are our challenges? And I think actually Nicholas already got this for me, but I think sampling is often a challenge. And also it may be hazardous for the people involved. So that's something I think we should consider. Sustainable funding is a challenge. We heard a lot also a couple of weeks ago in another conference, and also support from the public can be a challenge. The governance, of course, and public health interventions are not always clear, and especially integrating our results with other surveillance results can be also a big challenge. How many in the room are actually themselves epidemiologists? Oh, very good, very good. I'm impressed. Very nice. Because often it has been difficult to sort of integrate the epidemiology field with the other fields, like the environmental engineers and people that do this type of virus research. So coming back to the framework that I proposed on environmental surveillance for public health, I think the opportunities are in proactive governance. So really making good agreements with the partners. I'm so used to saying stakeholders, but I know we shouldn't use that anymore, but with the partners, and make good agreements on who's going to talk about which results when. A continuous cycle I would propose. So coming back from the public health actions, I think it should go back into the societal challenges. We need unity of language, and I think we should identify costs and benefits. I did see that that's an opportunity, or it's that we know the cost benefit, but I don't know. We're not so very much aware of our cost benefit, but I think we need more examples, or more examples published at least, to know this and be convincing. And then, of course, some general guidance that we've worked on with WHO, for instance, but also with the JRC in the European Committee in HERA. Working on different guidance documents, the cookbook, but also the environmental surveillance documents. And at the moment, we're with a group of experts, we're working on a draft guidance for prioritization and implementation of a multi-pathogen approach. So I wanted to flag that to you. And then last but not least, all the acknowledgements. Probably I forgot a lot of people that helped me understand all this. And if you have any questions, I'm here. I think our chair's not ready yet. Can I take a question, Yvonne? Or
Thank you. Thank you. Yeah, thank you. So we can have a question, the first question. Yeah.
So thank you very much for your presentation. Well, I made the introduction of the conference, but I'm a geophysicist, so I'm not really into that field, but I like your remark on ownership. When you say that, okay, some colleagues will be happy because they get a nice article at the end of the day, but you're horrified because this has some consequences. And for me, what you insist on is the maybe in some kind the missing link between the observation, the confirmation, and at the end of the day, who raises the alarm, who says, okay, this is serious. Because I think we may learn from other crises. For instance, there have been seismologists measuring seismic crisis, and they say, okay, well, that's interesting. Then afterwards, you have a huge earthquake with thousands of people really touched. And I think, well, I really like the idea to try to install some kind of active governance, because well, you may have observation, and then I really believe the correlation between observation here in wastewater and clinical cases, but we don't want to have clinical, don't we? So, and who takes the responsibility to say, okay, we have to enter something more serious and have vaccination, and how can it be done? On what kind of evidences, threshold?
Well, three things come to mind listening to your questions. I hope I can remember all three of them. One would be that we noticed that COVID was actually a splendid case for understanding how a really naive population was challenged with a new pathogen, and there was so much testing going on, and there was so much hospitalization data that we could gather, that this was a wonderful comparison, like from a data perspective, a wonderful comparison with the data we got from the wastewater surveillance. And I think my colleagues, the modelers in the Netherlands know much better than me, but what we learned was that also looking into different neighborhoods with a lower socioeconomic status, less uptake of vaccinations, less testing going on, that the wastewater surveillance was such a good way to keep an eye out for what was going on also in those communities. And I think learning from COVID, we should now take everything that we learned and move it into the future and see how we can use it. Some of the other things you raised, I mean, on climate change and infectious disease, I can spend another hour if you're interested. But there it also has been of utmost importance to measure a lot of data, for instance, on what was going on with fibriosis. So not per se Fibrio cholera, but also on parahemolyticus and phlonificus, and see if we could project, and by using models, project how much, based on the climate change scenarios, how many people would have more infections, have more disease, or would even die from those infections, to convince people that climate change and infectious disease are actually related. So to me, these hard data are very relevant to give us a good example. And I think with respect to the ownership, I had a point to make in response to your question. I think we all know that it's important, but it's such a different field, like with the threats that have been made, like making us feel less secure in our countries, in our capitals, in the other cities. I think it's very important to realize that when there's a threat, a security threat, at least in our country, it's the mayor of the town that is in charge. And so we should really pay attention to other types of ownership that we normally wouldn't think of. So these are not epidemiologists or clinicians, this is the mayor of a town. And what we struggled with a bit was in the Netherlands, we have these municipal health service regions, we have these provinces, but we also have safety regions. And in this is where we have to move. So this was a completely different group of partners that we needed to communicate with. So it's very important
and I think you have a really holistic overview, and some messages you had spoken here that it's beyond research. Yeah, we have responsibility to give answers to our community, and so it will be very welcome to have you with us for constructing and building up these guidelines. But what I would really like to say is it's not a question, but it's a comment about communication. It's absolutely pivotal. And so in Brazil, we had a very difficult political period together with pandemics. And in a period, our data was inaccessible. And so in Fiocruz, we have some databases and observatories and dashboards. But then we needed to make, in a very accessible language, reports. And so the press was waiting for our reports to inform the population. The population were assessing our sites to get it and to understand what was really going on in the absence of the governmental action, I would say, for that time. And so communication is so pivotal that must be carried out in different levels. I think first for community, because community engagement is everything. In every public health action we are going to propose or make. Second, stakeholders, because in fact they have the legal responsibility to respond, to implement, to assure resources, to really make it happen. And finally, the political class that usually we scientists do not like to speak to them so much, but they have the pen, they have the power, they are making the legislations. And so if you look at the pandemic treaty right now, the lack of consensus, why? So we must be, use the right language to speak to these different groups, because otherwise we are not going to establish anything. Yeah, we are going to stay inside our world, our scientific world, and what's needed to really face the real challenges we have now, not only for pandemic threats, climate change altogether. So the challenges are amplified. So we need everybody engaged. So thank you very much for your message.
Okay,
Perfect.
thank you. Well, I don't think I will go into politics, but I think one thing I forgot to mention was about the behavioral scientists, because we think that if the wastewater surveillance data would point at certain actions, then this is a very important field for us to actually have behavioral change, and otherwise we're less effective. One other point I was thinking about when you were talking about Brazil was that we were helping out colleagues in Ethiopia at the EPHI, our sister institute, and they told us that actually during the lockdowns, they were not allowed to go out to get the samples. And I think it was so important to have that information. So when you think about all these things that are not exactly data cleansing or whatever, or making nice graphs, but is that we should think about these circumstances. So now we can arrange for the next outbreaks or the next lockdowns or pandemics for these samplers to be exempt from lockdown situations. So think about it, talk about it with the policymakers. But I think I'm way out of my league now. Sorry.
Thank you. I think we have some questions from online people.
Yeah, from the chat. I'm here, Anna Maria.
Hey, hi.
Hi. So thank you for your great talk, say the participant online.
Thanks, Christophe.
So now, first question is related to the added value of wastewater surveillance for low-income countries. Is it sufficient considering the high cost?
Yeah.
And perhaps related to tuberculosis, given the chronicity of the disease, could it help in such condition?
Yeah. Well, I think maybe for that one, as you referred to my colleague who was just talking, but more in general, I think that's why I was pointing at these more simpler tests. But I think we should also not forget that, for instance, work done by the Africa CDC and empowering labs all around Africa. Don't be surprised if you would ever go there, if you have the opportunity. They have brilliant laboratories. So it's just try and get the electricity going and keep the consumables going. And but there's really brilliant scientists, and some of the labs have good infrastructure. So I think if we work together and open up a bit around the world, then we can help each other a lot. For the specific question on the TB, I don't know. Can you answer it better than me? Yeah, no. I think, for instance, around these types of diseases, there can be a lot of stigmatization. So where I think that wastewater surveillance may be useful is that if you have community sampling, then it's not important who is infected, but it's also important to know that it's going round. And so if you want to have targeted interventions such as vaccination campaigns, mopping up, or making sure that the right medication for the priority pathogens is there, could also be a refugee camp, or then maybe the stigma on these individuals could be a bit lower making use of wastewater surveillance. One other thing that comes up is that normally, as I understand, but I don't want to stand in the shoes of the WHO too much, but I think what's important is with these multi-pathogen approaches is that there's a national uptake, a local uptake of the priority pathogens. So you need to decide for yourself in your region with your public health nurses, your municipal health services, what is the top 3 priority pathogens in your country, or is it one that has your most interest and is most urgent? So you need to decide among yourself in that location what's most important. And I know lists are going around, but I think these really require reviews and stakeholder surveys. So thank you.
Another question? Maybe
I have
one.
a lot.
Yeah.
Okay. I have again some questions about the disconnection between the data obtained by sequencing for antibiotic microbes and the living strains.
Yeah.
So how can we deal with such disconnected results between both?
I think we also talked with our Danish colleagues lately, and also within the network of WHO CCs on AMR. I know that Frank, you're also, so your CC is also in that, but we had some discussions about whether to go for whole genome sequencing of isolates or to move to these metagenomic methodologies. But yeah, there is some discussion. So I did hear around the room to go towards these rhizostome profiling, but we had a lot of discussions because it's complicated, and also in relation to the plasmidome. So maybe we'll hear more about that a bit later, but we're not so sure that we should never isolate the bacteria again and do whole genome sequencing on the isolate. No. So.
Okay, so I think it's time to close now. Maybe one last comment on what you told us about the cold phase. It would be interesting to sample during the cold phase. It reminds me what Vincent showed, and that happens in summer 2020, where we could see in the wastewater that the epidemic was climbing up, and nobody was believing this because there was no testing or very few testing. And actually, we were about 3 weeks to 1 month ahead. So I don't know what to do so that there is more trust between what is done in the research for wastewater and the decision. What can be done in order to take this into account more importantly with alerts?
I think it's just very important to translate these lessons learned from COVID-19 into looking ahead and looking into influenza, maybe RSV. So yeah, move ahead and take those lessons learned and the whole validation that we could do during COVID and see whether there's early warning of the new influenza pandemic that is bound to happen. Yeah. Maybe not in our lifetime, but it's bound to happen. Yeah. Okay. Thank you.
So thank you again.
Yeah. Thank you very much.
The last session of this morning with the subtopic. So I have the pleasure to welcome our chair, Maria de Lourdes Aguiar Oliveira from the Fiocruz in Brazil. So the topic is entitled Adapting Wastewater Surveillance for Diverse Sanitation Infrastructures. The floor is yours.
So good morning, everybody, ladies and gentlemen. To begin, I would like to express my gratitude for the organizers to be moderator. I'm Lourdes Oliveira. I'm virologist by training and Vice President of Research and Biological Collections in Oswaldo Cruz Foundation from the Ministry of Health in Brazil. So as virologist, I work for the National Reference Laboratory for Respiratory Viruses and also WHO Reference Laboratory. And besides research, we make surveillance by mission. So I will stop talking about me because the most important are our speakers, and it's my great pleasure to welcome not only our distinguished speakers and specialists, but also all of you which will follow this discussion in person or online. So, why wastewater-based surveillance is not a novel concept, and since many years we use for different finalities that it was shown before. Its effectiveness as an early alert system, as well as a significant supplementary surveillance approach, was proved throughout COVID pandemic. So it shed really light. And then finally, it was a great opportunity to make it into a surveillance system. However, there are still many challenges, as we have seen in the speeches before. And I would like to highlight one special topic, especially in regions lacking sanitation. It's a huge challenge. Yes, so it goes beyond the only laboratory techniques or sequencing. So we have so many things still to do, but I would say that it would start by sampling how to do that. And usually these areas, it harbors particularly the most vulnerable populations. And so that's exactly where we have to act as a public health institution from Ministry of Health. And so I would stop by here. And during this session, our specialists will share their insights regarding challenges and future outlook of wastewater surveillance. And so I would like to invite Dr. René Street, Dr. Pierre Roque, Dr. Pilar Hernandez, Dr. Frank Aristrup, and Dr. Leila Anis Bulabal. And I will present each one in the order of speak. So I would like to present Dr. René. Dr. René Street is Director of Environmental and Health Research Unit at the South African Medical Research Council. With a background in both natural sciences and public health, she focuses on the impact of environmental containment on health. In response to COVID pandemic, Dr. Street played a key role in establishing the the South African Medical Research Council Wastewater Surveillance Program, which tracks SARS-CoV-2 RNA trends across South Africa and has expanded to include broader pandemic prevention, preparedness, and response research. So she will present to us the experience and the overview of this wastewater surveillance system and research programs. So Dr. René, the floor is yours. Thank you.
Hi, everybody. Thanks, Lutz. Lovely to be here to present on behalf of our team and on behalf of the SAMRC's Wastewater Surveillance and Research Program. I would just like to acknowledge the transformation statements of the SAMRC, which I stand by. So in 2020, many of you would know, many of us went into hard lockdown. The middle of, or towards the end of March, all our projects and programs, as we knew it, came to a halt while we were all looking towards a response to support the pandemic. We were looking towards the global north. We saw some really interesting studies coming out on wastewater surveillance. And we, in May of 2020, we did our first proof of concept to see if we could also show and prove the SARS-CoV-2 in our wastewater. Once we did, we were very confident in our approach, but we had not yet managed to see how we could do this on a larger scale. The city of Cape Town, where I live, there are 24 wastewater treatment plants, and over a 6-week period, we managed to track the wastewater signal in all of these 24 wastewater treatment plants, which really mimicked the COVID-19 clinical case data. It was really exciting for us, but we also knew that in a setting like South Africa and on a continent, there are many different ways that we'd have to approach wastewater surveillance, which we weren't going to learn from our colleagues in the global north. So in South Africa, we've got an extremely unequal society. We've got some high-functioning, usual wastewater treatment plants servicing the communities. And we also got some informal settlements with no sanitation. So this was me and my colleagues designing an environmental surveillance system to go and track the rivers around a community. Getting to that community, realizing we were expecting to see a river running through that community. And this is what we found. Really just showing the importance of getting out into the field to plan your studies and not doing it from your laptop, which many of us are guilty of. So through our response, after working with the city of Cape Town, we decided to rapidly expand our wastewater surveillance program, working with 6 partner universities through 6 provinces in South Africa. We used one method across our entire program and worked specifically with HDIs, which are historically disadvantaged institutes in South Africa. They were under apartheid era specifically for Black African students and remain historically disadvantaged today. They're under-resourced institutions, and we wanted to ensure that during our response moving forward, we were working collaboratively with these institutions. We provided, where needed, PCRs, sampling training, lab training, to set all these 6 partner universities up to move together with the same response. On a Monday, all of our sampling teams went out, and still do today. They go out to collect the samples across the country, bringing them back to their laboratories by midday so that the SARS-CoV-2 extraction can happen. On a Tuesday, the PCR analysis takes place and the quantification. And 4 years in, still every Wednesday, all of the lab teams meet together on one call at 9 a.m. in the morning to ensure that we're understanding the data, it gets signed off for QC, we investigating the local context of that week and really just understanding what's happening on the ground, but also we use this as a capacity building initiative with the different students and people on that call. After that, once the data's been signed off for QC, we then prepare different graphs and maps and different information packages for various key stakeholders or partners, and we update it on the SAMRC dashboard. We're the first in Africa to have a public-facing dashboard, which was very exciting. But as we know, this is not the only thing you can do to get information across. But we did this within 3 days so that for the Thursday and Friday, and obviously the weekend, it would give opportunity to key stakeholders and the health facilities to do the interventions required, either at local, provincial, or national level. At the SAMRC, we've got a state-of-the-art genomics platform at the beginning of the pandemic, we were couriering all of the wastewater samples to our one facility in Cape Town. This was very expensive, very demanding, and it was unsustainable. We used to do it weekly, then monthly, and now ad hoc as we can. We were very fortunate to have secured funding to now provide each of our local partner universities with a MinION that they can take action and also provide the training required at a local level to work with us on some of the sequencing data, which is really exciting. The science communication has been touched on in public communication. The pandemic was really tricky. I think one of the things that we learned on our program was really, I think we underestimated how to deal with all the different information coming across, all the dashboard that we had to set up. We had 4 iterations of our dashboard, how best to communicate the science to the public. In South Africa, we've got 12 official languages, so this also had to be taken into account. You can see here some of the feeds went out in some local languages. When we saw an increased signal in a local area, we'd put those out, trying to access as many different partners and possibles and communities as possible. But this was really quite a big task, which I think we underestimated in our planning. As we went along in this response, we wanted to ensure that the students that we're working with, the partner universities, were also being trained and understood the complexity of working in a pandemic. So not just training a student on a sampling technique or a laboratory technique. So they really understood, as we were learning and growing in our response, that they could also go along on our journey. So we planned writing retreats, knowledge mobilization exercises for these various students and partners to ensure that we all understood the complexity of what we were trying to do. Now, transport hubs are key to pandemic resilience. We've spoken, and we'll probably hear a bit more about airports and aircraft and monitoring. But in Africa, this is just 2 examples of CO2 emissions. This one is limited data on the flights, but it doesn't mean we are not traveling and not moving around. Because as you'll see, the second map, we are definitely moving, as you can see by the transports. And in South Africa, 55 million, there are 55 million minibus or taxi trips taken monthly. So we can also look at aircraft, but there are also other ways to look at these important pandemic transport hubs for pandemic resilience. And as we're looking at defined communities, we also need to bear in mind the ethical, legal, and social aspects of wastewater and environmental surveillance, which are lagging. I'm aware there's a panel later today on it, which I'm delighted about. There are currently 36 publications out on MPOX, mostly from the high-income countries. This one that I'm flagging here is the first one that I read that got my attention, really well-written paper that said, you can also go upstream and target undetected cases. But as we know, wrapping up. As we know, the MPOX is predominantly affects MSM communities, not limited to, but and in that response, 65 countries criminalize consensual same-sex relations. So this paper we wrote, Double-Edged Sword of Wastewater Surveillance, just really highlights that complexity and understanding the local situation when we talk about going upstream and define communities. In wrapping up, we have so many people to thank for such a dynamic program that we work in. We have so many obstacles, but so many wins that we celebrate at each step. For more information, please do reach out to me. Thanks.
Yeah, thanks, Dr. René, to highlight many different aspects. And I think it's extremely nice to see in the Global South, how do you really organize the wastewater surveillance? Because it's totally different from the Northern, because we don't have sanitation in most part of areas. And so how to act, how to do, how to organize, and especially highlighting the consequences, as Ana Maria was talking about, the consequences of the data we are generating. Yes, and so we must be prepared to give any kind of response in any aspects. So thank you very much for your presentation. So now I would like to call Dr. Pierre Roque. Dr. Pierre is a researcher from the Pasteur Institute of Guinea and had studied mother-to-infant transmission of HIV in France, African cohorts, as well as the HIV viral diversity in collaboration with Barré-Sinoussi Pasteur group. Thereafter, in Africa, Pierre had worked on epidemiology of retroviruses and hepatitis viruses in human and non-human primates. Back in France, he developed preclinical model in a non-human primate for HBV, chikungunya, and Zika that led to the first international approved vaccine against chikungunya. Wonderful, congratulations. Since 2021, in Guinea, he focused on detection methods, improvement to assess viral disease emergence, and survey through water sewage as a cheaper method than human infection diagnosis. So, Dr. Pierre, please, the floor is yours to talk about challenges of pathogen surveillance on, please, the presentation.
In Conakry.
Aha.
In Conakry area.
So the floor is yours.
Thank you so much, Maria, and thank you all of them to be here and the organizer to give me the opportunity to speak about our work in Guinea. I'm sorry. Ah, d'accord.
Oui, oui.
Ah, ok, pardon.
Le problème.
Ah, ok. Bon, ça y est, j'ai fait de mauvais points. Okay, well. As I have said, we will speak about the challenge of pathogen surveillance in Conakry wastewater. No. Yeah, okay. So just to remember that Guinea is a country in West Africa, which there are around 13 million inhabitants. And in Conakry, the capital, in the West Coast, there is around 2 million inhabitants. There is 2 seasons per year, October to May, a very dry season, and then June to September, the raining season with up to 4 meters of rainfall in this short period of time, which means that there is a sort of washout of all the wastewater system. And the temperature is always between 26 to 32 degrees in Conakry, night and days. I belong to International Pasteur Network through the Institut Pasteur of Guinea, which is the 33rd Pasteur Institute. He was born just after the Ebola crisis in 2014, and it became officially in 2017 as a Guinean institute, and not a French institute. I have to remember that very specifically. Our work is around the current and common mission of Pasteur Institute is public health, teaching, and research. And here we mainly focus on infectious disease. And then, because of the Ebola crisis, we work first on pathogen in animals as a risk of reservoir of this Ebola virus after the epidemic in human. And then arrived the first laboratory in 2018, and then the COVID crisis in 2020. And with the support of Pasteur Institute and other founder, we did a joint survey of the COVID epidemic by diagnosing the human population and performing the sequencing of the obtained the positive people, and we thus are able to follow the introduction of different variants from the here in 2022 with Omicron and the different variant of Omicron during the period survey here. And then in 2021, we have our first research building, and because of the go from the pathogen in animal to the pathogen in human, and now the point is about the environment. I just have to say that this diagnosis period of COVID was performed in collaboration with the Guinean Institute and Agence Nationale de Sécurité Sanitaire, the National Institute of Public Health of Guinea, and another and two other laboratories. One of them here, what are able to do both diagnosis, PCR diagnosis, and sequencing. And thus, we are interested by the pathogen in wastewater as a mean to detect and to follow the epidemic. So we first did a proof of concept by surveying the SARS-CoV-2 in wastewater in the end of the peninsula of Conakry, and the only area where there is a mapped wastewater network in Kaloum, and all this wastewater end to a reception tank here, then before the water was released in the ocean. And so we sampled the water here in this place, and also in the major hospital in Kaloum, in Yazdin here, to have a reference with a known infection in the hospital. And so we did that with two PhD students, and as everybody, we resampling the wastewater, we concentrate and extract the genetic materials, and did RT-qPCR for detecting SARS-CoV-2, but using commercial kits that are provided by the WHO and other founder for Guinean people. From this, we are first able to demonstrate that we can detect the virus easily in this water, despite the local temperature and the fact that most of the infrastructure was in a bad shape. But still, we are able to detect the virus and to correlate the number of positive samples in our reference site to the number of detection in the human population in this area, which here in the period of there are only a very few cases, just in the beginning of 2022, after the first Omicron diffusion in the country. And then here, for an example, in end on 2023, in the middle, sorry, on 2023, where they came to the last large Omicron diffusion, and in which case we are able to detect both in the hospital and also in the tank collection tank at the end of the network. And in this wastewater samples, we are able to sequence using immunion technology, and the detected subtypes was the same that we are able to detect in the patient in the same time, which is not a prevision, but just a follow-up of the epidemic. So by doing this work, we're able to implicate all the health services and authorities, and we have support from the INSS and also the National Institute of Public Health. And we participate to the weekly report about COVID epidemic, but also to measles, because there are current many outbreak of measles during the science to 2021 to up to now, and but also we are in demand by this same. We don't do it, but we are asked from the health authorities if we are able to check for cholera, polio, enterovirus, rotavirus, always in demand to increase our capacity, which currently we are not able to do, but which is important to remember that in this area, they need more information and they try to obtain that every way they can have it. So currently we are with the Atlant program, which is funded by the French Agency for Development, AFD. We implement our survey in the large Conakry. We've selected some sites because in this area there are not clear and mapped network of wastewater pipe, but only a stream, and we selected some place. And with some analysis of the population covered, we pulled this sample size and we did the survey. And because in this time we set up a team to do the sampling, and as you can see here, the situation is even worse than in the Kaloum area. That there is no protected stream, and all of them are full of garbage, and as I have said, often are washed out during the rain season, but the virus remain detectable even in this period when the washout is ended. And thus here it's just an example that we have done. Here at the time it was some cases in Kaloum, and but the most highest viral load was in this area in Ratoma. In Ratoma is a sub community here in Conakry. Okay, so the advance and remaining challenge of our study, and because, as I have said at the beginning, there is many outbreaks of measles, we did a retrospective analysis of the measles in the wastewater, and we are able to clearly identify the emergence of the current epidemic and in the end of the 2022 epidemic in 2030, and the beginning of the new one in 2024. And today we are able to localize in this area the place where we have also people that are detected positive for measles. And since then, because this is the data from May, since then we are able to detect the virus in quite every place in Conakry. The remaining challenge are as I have said, the large variation in flux of water and sewage, which means that we always have to adapt our sampling area with the situation at the time of sampling. Every 1 month or every 2 months, we have to check if everything is continue to be the same than the month before. The bad knowledge of the population data, because there are no clear data about the population. Always, we don't know the number of infants and the number of adults in this area, because buildings are always moving in Conakry, and there is a large population change even in this small area. And the point is to define the sustainability of the survey and sampling teams, because we already, after one year, we have changed half of our team to do that. And also, the next challenge is to have a better relationship to the health agency in order to set up the point. And then here to enrich the people who support our work, specifically OBEPINE, the founder and the Conakry Poubelle and the hospital in Conakry, and here our biology team, which has, you can see here, is a small team, and so we have mainly 4 people involved here, and here and here, and now these 2 also are involved in the survey. Thank you very much.
So, Dr. Pierre, thank you very much for your presentation and for highlighting some of the difficulties we do have when we are proposing to work in vulnerable areas. We don't have, the samples are too much more diluted, so it's a high difficulty to sequence afterwards. We don't have the catchment area, we don't have denominator for population, so we have really gaps of information. And so it's really challenging, and we need to improve how to approach wastewater surveillance in these areas. Our next speaker is Dr. Pilar Hernandez, who is one of the two managing directors of the Sorbonne Foundation. She has over 15 years experience working on programs for the control of infectious diseases, with a special focus in pandemic prevention and response, digital health, and neglected tropical diseases. She has provided technical assistance to Ministry of Health and regional institutions across several countries in Africa and Latin America. Hopefully Brazil will be the next, and led teams for effective implementation. She is currently responsible for the project's portfolio and business development at Sorbonne Foundation. So I would like to give the floor to Dr. Pilar Hernandez, who is going to talk to us about integration of wastewater monitoring and public health surveillance through Sorbonne. Thank you, Pilar.
Oh, okay, perfect. Thank you very much. I'm very happy to be here. Bonjour. I'm super excited about all the discussions today. So what I want to talk today, I mean, what we have seen today in the morning in all the presentations is the great potential of wastewater surveillance, all the technological advancements. But what we have seen as well is that many of these interventions are not yet fully and collaboratively integrated into the public health monitoring. And I would like to refer to one of the last comments by Anna Maria, where she said that public health as integration of wastewater activities might be tedious, like integration of wastewater activities into public health monitoring might be tedious. And I would like to respond to that maybe it's not. So what I want to present today is a tool that does exactly that. So I'm talking about the surveillance outbreak response management and analysis system, in short, SORMAS. So SORMAS, for the ones that don't know that yet, is an open source software. It is recognized as a global good. It does not only surveillance, the routine surveillance, but it has also epidemic and pandemic management capabilities. It has, of course, an event-based, it integrates event-based surveillance. It is used in a wide range of settings, including high, low, and middle-income countries. It has, of course, its desktop and mobile version. It can be used online and offline. Oops, sorry. It can be used online and offline. And I would like to highlight these last 2 points, because we are talking about adaptation of wastewater activities into different infrastructure settings. So when we have like health workers going into some areas that might be remote, we really need to have, they really need a system where they can collect the data in a way that in a place where the internet connectivity might be very poor or might be even inexistent. And these people will certainly use a mobile device. They will not go with their laptops and be connected to a Wi-Fi. So extremely important when we are using surveillance systems, they really need to be adaptable to the different areas. So the way SORMAS works, you should imagine, it's a digital platform. All the different users at the different points, being it at the community level, being it at the points of entry, ports, airports, etc., hospitals, laboratories, ministries of health, are connected to the same platform, and they share the information in real time. And here I would also like to highlight the real time, because there is no early warning without real-time data sharing, of course. SORMAS is not only a data collection system, and it's extremely important because it is not enough with collecting data. We have to do something with this data. So once everyone at the different work places where they collect the data, then they should do something with that. So the way SORMAS does that is that people, all the users in the different points, are able to assign tasks to each other. And this is within their teams or between other teams or places. So basically, you should imagine the data sharing is not bottom up, as typically many systems do. It is multi-directional and it's in real time so that people can also communicate with each other, coordinate activities through the platform. It has a laboratory integration, so the laboratory officers can enter the information of the sampling of the samples directly on the platform. It has several disease modules, and it follows the strict IT and security and data protection regulations, more specifically the European GDPR. So, following the One Health approach, an environmental surveillance module has been developed in SORMAS. And again, I would like to refer to what Ana Maria said, because I think it's important to distinguish the wastewater and the environmental surveillance. So this is an environmental surveillance module, which means that you will choose an environment, and in this environment, you can take different samples, being it water, being it soil, biota, etc. And within the water category, you will be able to select the wastewater category. So you will be able to collect all this information about the samples and also the parameters, being it pH of the water, volume, turbidity, etc., etc. Also, the results of the sample testing. So basically, we'll have your form, your sample form with all the information. And once the results from the laboratory are ready, then you will be able to integrate all the results of the pathogen testing in this form regarding this sample. It also has geolocation and a dashboard so that you know where the samples have been taken, etc., etc. We are now further developing this environmental surveillance module, and we are working on a dashboard where you will be able to see the human cases and the environmental cases on the same dashboard and geolocation. It allows to do the sample tracking so that you will see where the samples are from the moment that they have been taken in whatever in the wastewater location and to the laboratory. And this allows the real-time monitoring, again, highlighting the real time. So this way, what we have is in one system, we have the human health information and the environmental health information throughout the whole surveillance process, everything in SORMAS. So this starts with this detection phase where you have, as I mentioned before, all the different actors at the community, at the hospital, at the points of entry, etc. And also on the environmental side, it's just jumping, the environmental side. Then you will move to the investigation phase where the samples, being the human samples, environmental samples, are sent to the corresponding laboratories. And then data is investigated. So the sample data is analyzed. The investigation is done. And this is shared with the health authorities that are responsible for the decision-making on what are the public health measures, the right public health measures. So the advantage of having this in one system is that it allows data triangulation, because you have the information coming from the human side and the environmental side, which leads to a more accurate risk assessment and to an enhanced decision-making, because you have all this information, all the people working together on the same platform. So what are we doing next? We are going to pilot, we are going to work with Ghana on a pilot implementation for a national wastewater-based surveillance for multi-pathogen. Ghana is one of the countries that uses SORMAS as the national surveillance and outbreak management system. We will do this with the Kwame University and the Ghana Health Service. This is going to be done for several pathogens, including Salmonella typhi, hepatitis, measles, and pox polio. And this will be done leveraging on the polio sentinel sites that are already in the country. So I hope I did that in 8 minutes. So if you want to have more information about what we do at the SORMAS Foundation, here you have some links. You have the QR code to our website. If you want to get in touch, you have more questions. You have my email, my LinkedIn profile. So thank you very much.
So, Dr. Pilar, thank you very, very much for the presentation and to show us how to operationalize. It's because beyond everything we do have, how does we build up a real-time system, even with a stakeholder, to then activate the epidemiological surveillance to make the proper investigation? It's a challenge, especially depending on the political organization of each country, because the state has the province has a level of power, in all governance levels, I would say. So thank you very much, and it's very nice software. So next, I would like to invite Dr. Frank Arestrup. He was for many years the head of reference centers for AMR, for European Community, WHO, and FAO. And since 2009, he was pioneered the use of online bioinformatics services to promote the use of next-generation sequencing, especially in low and middle-income countries. During the last 10 years, he was explored how metagenomic analysis of wastewater can be used to perform surveillance of AMR and pathogens and possibilities to assist countries in implementing and sharing data globally. So I guess there will be thousands of questions in the chat for you afterwards. Be very welcome. The floor is yours.
Thank you. Well, I don't know if there's going to be thousands of questions, but that will of course be wonderful. So just arrow right up or down. It works. So yes, as said, this is well, we've been using metagenomics when we started looking at approaches of doing actually not wastewater, but this was animal feces surveillance, looking at individual genes. We actually did explore very early on qPCR, but we very rapidly felt we had to go for metagenomics, because, well, basically we had around 3,000 targets we had to look for. So it was just not feasible to even to start thinking about qPCR. And I think that perhaps that's also going to be the case in all the rest of the surveillance, that as the number of targets increases, well, you'll probably have to redirect away from the qPCR. And I'm afraid that those of you that are doing qPCR, you might also explore some challenges when that's going to happen, or you might just be smarter than we were, because and overcome these challenges. We started out actually looking at long-distance airplanes, but very rapidly decided we need to go out and see how can we explore this in a global context. Also because when it comes to AMR, Denmark is actually quite boring because we have a relatively low level of resistance. So we did a small pilot project back in 2016 to see how could we actually collect data or samples from around the world, ship it to Denmark, and then, do the analysis there. So very much, the good old-fashioned send a sample to us, and we will do the analysis, something we really need to get away from as we move forward. In this case, it was 79 locations from 74 cities in 60 countries. And I think that what we saw, at least then, was that perhaps a little bit as expected, the abundances of it, the resistome was highest in sub-Saharan Africa. But we also saw what was a bit challenging for us to understand, this separation the world's regions into different, looking at, the PCA plot in the lower left corner. One thing we also did at that time was to say, okay, let's see if we can correlate this to some data, some other data there. I actually expected that we would see almost perfect correlation to antimicrobial usage and then the abundance of resistance. That was not the case, and we actually had a hard time getting that correlation, significant. There was also a lot of lack of data on antimicrobial usage for most of these countries, but nonetheless, we had challenges there. But we did see correlations to, parameters for the health system, so like number of medical doctors, water and sanitation, the WHO Human Development Index. There was actually pretty good correlations. So we explored further all the World Bank parameters and adjusted, machine learning, random forest, and see, okay, what is actually predictors of more AMR. And it actually looking at it, it seems to make sense. This is biological drivers. This is not the number of gold mines or electric, conductivity, the level above the sea, number of mountains regions, and so on. It's really parameters that actually seem to make sense. So just for us, I think that we also confirmed this in other studies, and there's also been other confirmations. I think that we need to see antimicrobial uses, in the context of socioeconomic factors, because of course usage selects for resistance, but it is in a context. It is not just by itself. And I think we need to understand that if we want to address AMR in the future. And then of course we can make predictions for the world as the map we just have there. We continued this and expanded it to 757 sewage samples from more than 100 countries, and collected over some time. More time. I think what we saw was, confirmation of our first findings that we could really see this, regional separation in the resistome, something that has puzzled us a lot and actually still do. But we could also, when looking further into the details, we could see that this regional separation really is very dependent on the classes and even of the exact variant of the resistant genes we are finding. So I think that perhaps we also need to come to grips with that out of the more than 3,000 acquired resistant genes we have, they are probably each of them following their own global evolutionary trajectory, and understanding all of it is probably going to be a challenge. But again, this is really where we need to aim if we want to solve this. Because, AMR is not just AMR. AMR is a lot of different resistant genes jumping around, and they need to be addressed and understand properly individually. However, also having these technologies in place, we have also said, okay, can we also use this, in other contexts? In this case, 5 different European cities where we have done longitudinal sampling. I think that, at least for us, interesting concept here. This was, by the way, published relatively recently. The interesting concept here for us was that, well, first of all, we actually couldn't go out and say there's a lot of things to discover out there. There's a lot of unknowns. So in this case, more than 1,000 novel taxa we have been identified by metagenomic binding. We have projects now going on in Africa where we, at least initially, could only assign 2% of the bacterial reach. So just imagine that you take a sample and 2% can, only 2% can be assigned to known taxa. Then you do metagenomic binding, and we can increase that to around 90%. But there's a lot of discovery, especially in sub-Saharan Africa. And I think that there's a lot of things we are missing there. The most important thing here perhaps is that basically for us, we can see that if we have around 10 samples, we have a sufficient of a unique signature per site on the bacteriome to actually, well, then sample number 11, 12, etc., we can assign that without having the metadata. So as soon as we have a number of samples, well, we do not even need to know the geolocations anymore, because there is actually quite a good unique signature per sewage site. That's, of course, still needs to be examined. Doing network analysis, it also seems that we can get the bacterial taxa together. There is a lot of variation over time, of course. There's a lot of other bacteria that have nothing to do with the fecal contamination. There's a lot of growth in the sewer system. But doing these network analyses, we actually do believe that very soon in the future, we can identify the fecal, population in this. So basically, the 15%, so 15% that are, the fecal contamination in these, on average in these. And then we can start linking the antimicrobial resistant genes and perhaps only look at those that are relevant from humans and not what is happening in the sewer system. I think that will come relatively soon. As a small, digressions on this, one of the things we also saw when we started doing these bindings was that we actually can start detecting pathogens or presumed pathogens even in low abundances. We are not sequencing that deep. That will probably have to be, we have to sequence more in the future, I think. But we have seen a few reads mapping to Vibrio cholera for a long time in Denmark. Not something I have just ignored it. This is false positives. Who cares? It was then confirmed by IBM in the Netherlands by qPCR, and we said, okay, let's revisit this. And then looking at 115 samples, we could actually get a complete genome. But we could also then identify that, yes, it was Vibrio cholera, but they did not have the toxins. So we actually showing the relevance and the value of being able to, reanalyze the data when you have them. And again, I think that speaks in favor of metagenomics as and having the availability of the data sharing in the future. A very low abundance, as it's 1 out of 500,000 reads. We had the abundances. The global sewage has continued until we are stopping it. We have more than 1,000 samples, 111 countries. Now we are actually analyzing more than 14,000 antimicrobial resistant genes because, yes, there are these resistant genes that are known to jump around, but a lot of resistant genes have been identified by what we call functional cloning. They can cause resistance. They have not emerged into clinical pathogens yet. So looking at this, we try to say, okay, let's see if this changes the pattern. We confirmed the clustering for the acquired resistome, but actually, when we look at the functional resistome, we can see that they are basically everywhere. So in other words, we have just as much, functional resistome is this room as if you took a room or any other setting in sub-Saharan Africa. So we do not see the same abundance and diversity change there. When we look at the gene variant level, we do see a very clear classical distance decay. So as we also see in ecology, so the further you are, there's simply the less similarity you have. So we have a similarity drop. That's continuous for the functional resistome, but actually it simply levels off when you compare between regions for the acquired. So there's something happening there that you have a regional signature and you have a distance decay within regions, and then there are some genes that are jumping globally. At least the acquired ones. Whereas the functional resistome, there is this decay all around the world. If you have other good suggestions for this, then fine. But I think that for me, it also shows the value of actually having access to relatively large scale data, because then you start also seeing, global patterns. So concluding remarks. Well, I guess they're preaching to the choir here, but surveillance is the basis of everything, and without it, we are flying blind. And I still think that we are very far from actually having implemented a global surveillance where we are also doing much better in terms of sharing the data and sharing the raw data with each other. And qPCR is great, but as the number of targets increase, I think that we already, but I think the world will have to look at other approaches. Metagenomics also allows for discovery of novel species as well as reanalysis. And then, of course, data sharing is essential, but we need to find a model where we can actually protect, the data owner and create a global equity. So it's not just, okay, the Europe and North America will take all the data and analyze it for you and get all the credits. I think that there needs to be some models where we are also providing the data generators, with abilities to analyze, share, and control the data. And I think that we need new models for that. Thank you.
So, Dr. Frank, thank you very much. I must confess that I have envy of this huge study and the cost you should have taken. It's really amazing, and we really do need this kind of broad picture. And so along your presentation, first, I think you have highlighted the social determinants of health. It's extremely important to have in mind, especially depending on where we are and each country, each region, each kind of population, because even in the rich countries, we do have the vulnerable populations, and the social determinants are totally different from the rest of population we are dealing with. Second, the real power of metagenomics. It's amazing not only to detect emergent resistance or pathogens and so on. And thinking about pandemics, it's absolutely important because it's the best way we do have now to really see that. Otherwise, we are really blind, as we're telling. And so to tackle epidemics and also to make this big map of what do we have, and then you are able to prioritize where to invest your resources. That's absolutely brilliant. The single thing, and I will put fire for the next discussions, is as the number of targets increase, maybe metagenomics is more feasible, but in terms of costs, not maybe for the global south, not because the taxes we are paying is still very, very expensive for us. Yes, and afterwards, the other part that we will comment later is about bioinformatics. It's not an easy task to really make interpretation of data, so that's something that it will be wonderful if we could discuss afterwards. So thank you very, very much for your presentation, and now I'd like to call Dr. Dabia Leila Anis Bulabo, and I'm very sorry if I'm not able to pronounce your names right, so you can correct me. And so Dr. Leila is the head of antivirus laboratory at the Pasteur Institute of Algeria. She is an expert in biorisk management for the African region, and her laboratory is WHO National Reference Lab for polioviruses, including human and environmental research and surveillance. In 2007, she initiated and led environmental surveillance deployment for enteroviruses at a national level and has contributed to the supervision of environmental surveillance sites in the African region for the WHO. So Dr. Leila, be very welcome. The floor is yours. She will talk to us about multi-pathogen environmental surveillance in the Algerian experience.
Thank you. Dear colleagues, thanks a lot for giving me the floor and to participate to this very important event in behalf of my institute. I will present, the multi-pathogen environmental surveillance in Algeria. We initiated the environmental surveillance of wastewater in Algeria in 2003 as part of project with Institut Pasteur of Paris for enterovirus surveillance, especially polio surveillance. We are part of WHO Global Polio Laboratory Network for environmental surveillance in 2017, and we have polio sites. We do systematic surveillance of SARS-CoV-2 since October 2020, rotaviruses since November 23, and recently adenoviruses. We have 9 sites from the north to the south in 6 provinces, and the collection schedule is 1 to 2 samples per month, depending on the epidemic situations. And we use the GRAB method for the collection. For the method for polio, enteroviruses, rotaviruses detection, we follow the WHO SOPs and the guidelines on environmental surveillance of detection of polioviruses, and we concentrate the virus using the polyethylene glycol 6000 and dextran treatment with chloroform. And after that, we isolate the virus on cell culture. We confirm all the polioviruses using enterotypic differentiation by RT-PCR, and we genotype EVs and rotaviruses by RT-PCR endpoint, and all the positive are confirmed by sequencing. There is no virus isolation for rotaviruses, is directly RT-PCR. For the result, a good site for, according to WHO, is a site who has up to 15% of EV detection. And in the distribution of enteroviruses, including polio, in 2024 across all the sites in Algeria is 99% of enteroviruses, and all the collection were done in the inlet of the wastewater treatment plant as recommended by WHO. In our previous study between 2008 and 2012, we had 97% of EV rate, but all the samples were collected in the biological treatment basin of the wastewater treatment plant. This to show you, to demonstrate the resistance of non-enveloped polio or non-enveloped viruses in this rude environment. The impact of the time of collection. In considered sites, the time of collection is between 1 to 2 p.m. according to the peak of flow entering to the treatment plant. And we see that in 2017 we had 100% of EVs that was isolated in that site, and after that in 2018 15%, and in 2019 only 15%, which is very, very, very bad rate. We supervise this site and we did our investigation, and we have the information that the population size has increased in that region, which has led to a change in the timing of the incoming wastewater flow to the treatment plant, shifting from 1 to 2 p.m. to 10 to 11 a.m. And the recommendation is to do parallel testing between the two times of collection in order to compare. And in 2020, we did that comparison, and in the oldest timing of collection between 1 to 2 p.m., we found 46% of EVs, and for the new timing between 10 to 11 a.m., we found 77% of EV rates, which is very good, and we since that we adopt this timing. This is to give you information that when we have a bad site or a site which have a lot of negative, we have done to close it directly. We have to investigate in order to know what is the problem in that area. For rotaviruses, as expected, and like enteroviruses, we have a big range of positive for rotaviruses from the north to the south. For SARS-CoV-2 detection, at the beginning of the pandemic, of the COVID pandemic, we use the archived concentrate from environmental surveillance of wastewater sample of poliovirus for COVID detection, and all that samples were treated with the polyethylene glycol 6000 and treated with the chloroform, and we processed directly our RT-PCR. The result was not very good. We have some difficulties to interpret. We had a lot of flat curves or barely raised curve with one gene instead of two. This is a big issue because despite of the pandemic wave, the ES detection of SARS-CoV-2 remained negative, suggesting maybe an inhibitory effect of the chloroform on the detection. We concentrate alicot, and we concentrate again, and we take alicot before treatment with chloroform and after treatment with chloroform in order to compare the results. And we did our RT-qPCR, and the result was that all the samples treated with the chloroform remain negative or difficult to interpret. And for the samples who were concentrated without chloroform, all the samples are clearly positive. How to clarify doubtful results? We try to adopt other techniques for SARS-CoV detection using the concentration with the polyethylene glycol 8000, and all the negative result with PEG polyethylene glycol 6000 without chloroform, but negative or difficult to interpret were positive with polyethylene glycol 8000. And for SARS-CoV-2, this technique was adopted since. This slide is to show you a positive result during the COVID wave without concentration and without extraction. Is directly the wastewater with primers and probe and direct PCR. For the predictive effects, before the Omicron emergence, this result show you the evolution of the viral load between October and December 2021. And in October, as you see, we have a positive result with a very low viral load. In November, the load is a bit higher, and in 6th of December, the load were very high. And in the mid of December in Algeria, the COVID wave was confirmed. For SARS-CoV-2, the detection is positive across all the sites with different ranges from the north to the south. In the south, we have 44% of positive only, but maybe it's related to the high temperature in that region. As challenges and conclusion, we have to optimize techniques while waiting for standardization, because in the beginning of COVID, we were facing to a pandemic without knowledge about the techniques in the for detection in wastewater. We have to ask the question of the reuse of the wastewater after the treatment plant, because if there is a pathogen virus like rotavirus, poliovirus, which is a live virus in the wastewater after treatment, what is the policy of the reuse of the wastewater for agriculture, for example? The polio environmental platform can provide an excellent opportunity for other studies to be undertaken and may require multidisciplinary team for do this job. And thank you for attention.
So thank Dr. Leila to share with us your experience and the power, how surveillance, how a reference laboratory can improve not only our techniques and protocols, but how we are adding value to surveillance investigations. So in brief, along this panel, this discussion, let me only put the glasses. The letter is already horrible. Without glasses, I can't read. So we had seen the approach of wastewater under the One Health perspective and its effectiveness into surveillance framework. We had spoken and discussed about ethical implications, difficulties of many different natures in vulnerable areas. And so how are we going to think about that and to build up a better approaches? Sustainability, that's absolutely essential to not only to maintain what we had built up, but also to improve and to add new pathogens into the surveillance scheme. The operationalization of this surveillance, and so how to do under a multi-pathogen fashion. As the software we had seen, it's an open software and so on. And afterwards, you can speak more about how to access and something like that for those who is listening to us. Metagenomics, and so the power, yes, and how we can make its use more usual at the wastewater samples, because then we have really a populational approach instead of the clinical laboratories which enter the clinical cases. And we have so many gaps because of this kind of approach. And so when we are connecting into the surveillance framework different strategies and approaches, then we are really going to make early warning, and it's essential for tackling pandemics. The social determinants of health and its importance to really understand and help us to interpret what we are finding at the wastewater, the influence of concentration methods. So it makes all difference. And so we must really know better about that for each pathogen and how to standardize the protocols. Then that makes all the results, especially when we look at under the global view, to make results comparable. in the reuse of wastewater. That's absolutely critical because as time goes by, it's more and more used even for agriculture activities. So I would like to thank very, very much our speakers for the brilliant presentations, and would open for questions from the audience who is in person and who is online. So please, you are free to make your questions.
Hi, my name is Gildas from University of Copenhagen. I have a more technical question. It is to Frank. Thank you for the very nice presentation. You've been, I think, at the forefront of the wastewater metagenomics field. Again, it's a technical question. We have noticed that it's always very difficult to link the antimicrobial resistant genes to the host bacteria. And with the advent of binning, constructing mags, everyone thought we would then be able to do that, but it's still been very difficult for the genes to make it to the beans. They usually make it to the assembly, but later when you start refining your beans, then you lose them. So I know you have a lot of developers in your team. Are you guys working on new tools for better binning modules so that we could make it easier for the resistant genes to get into the beans instead of doing statistical correlation?
Yes and no. I don't have a solution if that's what you're looking for. We are trying, we have tried, high C and single cell sequencing. And of course, it looks very, very good on paper, and it looks very good when you look at the publications, until you actually read the details and say, oh, they only have 15% complete bins or something like that. It's difficult with both of these technologies, even though they have promises and they are extremely expensive. We actually have a study coming out with high C very soon. I think that for the global scale, I still think that the network correlations, which is the statistical correlations, that is going to be what is going to be closest to in the near future. So I think that networks is where I would go right now. And now, if I have it, I just have the opportunity to. I'm sorry, yeah, but low middle income countries. Yes, I disagree. I think that for some of these countries, it will be much easier to set up metagenomics and do that. And you can do it for AMR, get relevant data for less than $100 per sample. And also remember, you do not need a lot of equipment. You have your MinIron, you have a laptop, you have a bento lab, and then you're in business, and you can move anywhere. And whereas I think the bioinformatics is really, where it's going. We are not going to be able to educate sufficient number of bioinformaticians in the near future to have bioinformaticians everywhere. So I do think that online availability, freely available, is going to be really, really, really needed. And I know I'm talking, we have some tools we have developed. There are also others like, the James Zuckerberg Foundation. Their ID pipeline is actually also pretty good for metagenomics. So it's also using our aligner. So I have to, I'm a bit biased there, perhaps.
Thanks.
Hello, everyone. Valerio from University of Milan. I'm a postdoc, and I have another question for Professor Arestrup. I'm also a novel bioinformatician and data analyst, so I'm really happy to see a lot of data explained from you. I have a technical question too, maybe. First of all, the platform, maybe I didn't get it, but what platform for sequencing did you use in your work? It's Illumina or long reads for nanopore? And the second one is mainly related to antimicrobial resistant genes detection, meaning that there's a lot of controversy in literature about the minimum number of reads to say that our sensibility to detect all the genes in the sample is enough, let's say. Yeah, thank you.
Illumina for all of this, and this is to make the data comparable over time. We do add now with long reads, and I do personally see that that's where that the technology is moving. This is the future. It is simply cheaper. I do not have stocks in any of the companies. It's cheaper and more simple. So I think that's where calculating the abundances is a little bit more complicated there. I think that's and the number of reads for sensitivity. I think this is basically where I was just writing an email today suggesting to our public institution in Denmark that perhaps we should simply do a comparison, running a real time, comparing metagenomics and what they do. And also looking at the cost effectivity and sensitivity. And I think that the rest of you should do that as well. Exactly there, we do actually need studies, and we have a lack of a black hole right there.
Okay, I had a question for Pilar Hernandez on Restore. The question is about the right part of your table, so the decision-making and the control, how much of that you have implemented in Restore. So we saw you have a lot of the stuff on the left about surveillance. Do you have tools for helping decision-making and for somehow simulating the control measures to help the decision-making? Or do you plan to include that in Restore?
So at the moment, SORMAS it's not, I mean, decision making has to be done by humans. So the thing is that, of course, so the idea of SORMAS is that you can use all this information coming from SORMAS, collected through SORMAS, like analyzed through SORMAS and so on. Also, like, predictions, I mean, it's something that I have implemented yet, but it's certainly possible, like with machine learning, like outbreak prediction. But at the end of the day, the decisions had to be made by humans, because there's no software that can replace these decisions. So no, you can simulate things, but like the decision has to definitely be made by humans.
Fortunately.
Thank you very much. Thank you very much for this wonderful panel. So we had an example of Denmark, Guinea-Conakry, South Africa, and Algeria. And this is the first time I'm part of a discussion where we discuss site selection and up to public communication and risk communication, all the way through laboratory and all those technicalities, ethics, all those topics. And we want, and to my knowledge, my very small knowledge on wastewater surveillance, the only example that I have is polio, for which we can have comparable and we can compare results across countries. So where do we start if we want to expand to other pathogens and different settings?
Where do we start? As you said, the demonstration was done with polio, and this is a start for wastewater environment survey of pathogens. But the point is, as pointed out by Leila, that we have to also set up the concentration and extraction techniques. So we have just back to our basic knowledge of virology. We cannot use the same system with an enveloped virus and a non-enveloped virus and also bacteria. So we just have to, perhaps one of the first steps also at the end was to provide not a single system of extraction, but a panel of different technique or procedure, in fact, taking into account both the virus, both the environment, both the and also the temperature and one of the points we have specifically, for example, in Guinea, we did only in Conakry, because just for transportation of the sample from the inside country in Guinea or Guinea Forestière, we need it's by car. It needs 6, 12, or 24 hours. So we have to take all of this in account, and also the fact that
perhaps one of the points we have is also to provide some technicals we are able to transport with at ambient temperature, as we have done in Conakry, in fact. So I think the base is to provide not a single method, but a set of methods, and with a particular focus on the various un arbre des décisions. Sorry, a decision tree that we take in account of this point. And also at the end, I remember about the communication tools and the informatic. So for the presentation, and also we have also to remember that in each country, the health services use a specific information system. For example, DHES2, which is very efficient and largely distributed, but it is not always very friendly for wastewater results. And also we have also to set up what is the right windows, 1 week, 1 month to give back the data, and which is also the same close to the difficulties to have where have we to sample once a week, twice a week, once a month. It's also will depend of the pathogen and the dynamic of the epidemic you will follow up. I remember that about chikungunya, for example, the epidemic will be started in less than 2 weeks, and they have a peak in 1 month, and everything will go down, like in La Réunion Island more than 15 years ago, and in the other country. So the timing is very different. Just a point. Okay.
The last question, or maybe the virtual question, because we must close the session, and I would like to give 1 minute for each speaker for the final words, final considerations. Is there any virtual question?
Yes, we have some questions here on the chat. So few of them related to the areas that are difficult to reach, or someone is asking about humanitarian settings like refugee camps or war areas. Is it reasonable? Is it you think it's applicable? Because it's also regions where populations are endangered by infectious diseases. So is there an added value of wastewater surveillance there, and is it doable?
Yes, and yes. So I think that absolutely it's well, you can go out, you can take a sample, you can as I said, I'm very much in favor of metagenomics because it is extremely flexible, but of course you can also analyze it using other methodologies if you want. And it's available, yes, absolutely, because of course you want to know what is circulating out there. You perhaps even want to know what is some kind of a resistance pattern that is actually happening. Right now we are, as a cabinet, we are also looking into the metagenomics of these, mpox metagenomics. Can that be helped, to direct, treatment? Is there something that can help in these really, really, really poor and difficult to reach settings? So yes, absolutely, I think it has value. And just to address, where do we start? I do think that each country has to decide, what they want to go for. I don't think that it's not up to me to dictate, you do this and this and this. Different countries have different problems, different starting points, different funding models. So I think that it's up to them. I would speak very, very, very favorable of metagenomics, but it's really up to the countries what they want to do.
So thank you very much. I would ask you for the last considerations, please, 1 minute each. Who wishes to do that?
Okay. So yeah, thanks. Great panel, everybody, for the team. I think through my closing remark and just hearing everybody speak is with the climate crisis ongoing, changing rainfall patterns, frequency of extreme weather events, our systems really need to be designed around agility, and not only, the pathogens changing over time due to these the climate change, but also how we access samples. I mean, a whole wastewater treatment plant could get wiped out during an extreme weather event. There may not be water to collect the sewage water. So we really need to think about this going forward and the agility of these systems that we're designing, right from the sample to the endpoint. Thanks.
Yeah, I can continue. So I think one of my main points or considerations will be here that it is extremely important that we break the silos. We should be integrating our activities. We have people working on the human health, we have people working on the environmental health, but at the end of the day, what we want is to protect the health of our population. So we really need to, and when I say we need to break the silos, I'm not referring to something in theory, I'm referring to finding some practical examples, creative ways in which we can collaborate with each other and then have a higher impact. And again, in the morning, there were some people mentioning about funding resources. So if we work together, we would try to break the silos practically, then we will be using our resources more in a more intelligent way, and we will be increasing our impact. So I think that will be my takeaway message. And we don't need to start from scratch. So there are already people working on that. Thank you.
So, well, I think that for me it is that we do not know where the next problem will emerge in the world, and we do not know who's going to generate those data. So I think that we need to enable more people around the world to not only generate the data, but also analyze the data and share it. And I think that also we do need to see public health institutions being a little bit more adaptable to actually take up data from other sources that they have generated themselves. And that's going to be a challenge. But I think open data sharing is really the key, and we need to facilitate that.
Dr. Pierre, Dr. Leila, yeah. No? So thank you very, very much for the discussions and all the shared experience. So I pass the floor for the organizers if they want to make any kind of communication about the lunchtime.
Dear ladies and gentlemen, I propose that we start our afternoon session on time. So this afternoon session is dedicated to from research to policy. And I have the great pleasure to welcome our chair, my colleague from Panthéon-Assas University, Simon Porchet. Welcome. Floor is yours.
Much. I'm going to use this mic because this is a round table, so it's more informal than maybe the other sessions. So hello everyone, welcome to this session on this roundtable actually on ethics and legal aspects of wastewater-based epidemiology. We saw this morning different applications of wastewater-based epidemiology, and we saw that it has a huge potential to change the life of many people, but alongside those benefits, it raises also many issues, ethical or at least questions, ethical questions or pressing legal issues that we are going to discuss today. I'm Simon Porchet. I'm a professor of management at the University Paris Panthéon-Assas, which is a member of the 4EU+ Alliance. And I'm glad to be on the stage with three persons that are going to discuss those ethical and legal issues. We have Natalie Ram, who is a professor at the University of Maryland at the King Carey School of Law. Talia Bickman, who is a program manager at the Canadian Water Network, and Jean-Baptiste Guillonnet, who is an associate professor, of law at the Panthéon-Assas University. They'll provide different insights on three main topics the legal frameworks of wastewater-based epidemiology. We will discuss also some ethical guidelines of WBE, especially on the issues of related to data. And then we'll explore different topics, privacy challenges, community engagement, and other issues that are related to ethical and legal aspects. So thanks for being here, and let's dive into the discussion. We'll start maybe with the legal framework, just to have a sense of what's going on in the United States and in Europe, because we have the opportunity to compare the two different models. So what are the main laws governing the collection and use of data from wastewater-based epidemiology systems? Maybe we'll start with the US, Natalie, if it's fine for you.
Yes. Yeah. Hi. Can you hear me all? All right. Wonderful. So I'm delighted to be here and in this conversation. So thank you. You know, when we compare privacy and data protection law in the United States and in Europe, the United States doesn't look so great because we don't have a general privacy or data protection statute nationally in the United States. There's a lot of clean water regulation, but that's generally looking at the outflows of what's leaving the water treatment plant. And there hasn't been a lot of regulation on what you can do with material flowing into a water treatment plant, right? And so we could use some regulation in this space, especially now that wastewater-based epidemiology or wastewater surveillance has become so much more popular across the United States in the wake of the COVID-19 pandemic. I will note that one of the great benefits, at least in theory, of wastewater surveillance is that it is genuinely aggregate, that everyone with access to a sewer, right, deposits their contribution and it all gets mixed together. And in theory, you shouldn't be able to identify what I added from what you added, right? And that really tends to attenuate, to really minimize our concerns about privacy. But the farther upstream in the wastewater stream you get, and the more granular the data gets, and the better the science gets, the more likely it is that we're going to be able to disaggregate the data or identify specific individuals' input into the system. And so I think there is some space to think about the implications for privacy. And so, then I will wrap up. When we're thinking about privacy, I think in the United States, we think about privacy vis-a-vis two different types of actors. If we're thinking about privacy with respect to the government, then we are thinking with respect, say, to law enforcement, how law enforcement is gaining access to private information or sensitive information about us. Then we think a lot about the US Constitution's commitment to bar unreasonable searches and seizures and other regulations that have been put in place to regulate how law enforcement does their job. But generally, privacy statutes have an exception saying we're not touching law enforcement, right? When we're thinking instead about privacy with respect to private entities, the United States has taken a model of notice and choice, right? So there are terms of service, nobody reads them, you click the box, and you've consented to whatever's in that document and they can change it at any time, right? So that would be like privacy online, but perhaps also, I don't know if there's a terms of service to my water utility. I've never checked, right? And so those are kind of the two frameworks of privacy.
Thank you. Maybe Jean-Baptiste, if you want to have a slight comparison with what's going on in the EU, for example.
Yeah, thank you, Simon. I believe WBE data collection raises new juridical question in Europe relating to human rights and privacy. About data protection, the main legal framework is from GDPR, but not only because the Council of Europe has a Convention 108 and the Article 8 of the European Convention on Human Rights, formulating the right to respect for the private and family life, can also apply. The GDPR enforcement is based in Europe on a logic of accountability and of a high level of human rights protection. So the question is about fundamental rights of a natural person. And the key issue about WBE data collection, I think, is about the ability to identify a person, either directly with the data, but also in a large interpretation with cross-referencing. A point is really important about it. I think, and the explanatory report of Convention 108 precise this point, that an individual should not be regarded as identifiable if his or her identification requires unreasonable time or effort to identify. So I think a general wastewater monitoring does not permit to obtain data relating to air, to health, or to an identifiable natural person. It would be different, and it would be different concerning relating to a little scale, such as university or airports, if WBE data collection is associated with cross-referencing of cross-checking, such as medical information. But GDPR does not prohibit sensitive or health data processing. Just you need a legal basis justifying data collection, whom consent, legitimate interest, public interest, or legal obligation. And I have to notice here that there is currently a proposal of a new directive, a request, about wastewater, urban wastewater. And this proposition introduced an Article 17, which enshrine the requirement of a national surveillance system based on urban wastewater relating to different viruses, including SARS-CoV-2 or emerging pathogens. Thus, this legal obligation will justify general wastewater monitoring for this purpose with a flexible legal framework without consent or opt-out for the people. Maybe it will be slightly different about drug conception, for example, but we can speak about this later. Last point about Article 8 of the EHCR. This article authorizes interferences in private life, but the law permitting this has to be really clear and predictable and necessary in a democratic society.
Thank you. Let me open the discussion to ethical aspects with Talia. So Talia, can you just remind us also what you're doing more generally at Canadian Water Network and summarize, let's say, the ethical guidelines that you derived, because it's very interesting. It's different from what you can see in research because it's more practical. So we'd be glad to have an insight of the guidelines that you wrote at Canadian Water Network.
Great. Can everybody hear me? It's on. Hopefully everybody can hear me. So first of all, at Canadian Water Network, so as a non-governmental organization, we aim to tackle various complex water issues through convening decision makers, things like developing communities of practice and other knowledge mobilization activities. For me in particular, I'm an epidemiologist by training, and I implement CWN, Canadian Water Network's peer learning program for wastewater surveillance. Hopefully you can hear me now, and hopefully I was projecting enough that I could be heard in the room. Apologies to those online. But yes, so I implement CWN's community of practice for public health practitioners in Canada, focused, oh, there we go, okay. Okay, it's focused on Canadian public health practitioners, aimed around knowledge sharing, peer learning, and just developing communities of that community of practice and knowledge mobilization resources. So specifically with these ethical guidelines, this current program, this peer learning program that I implement was born out of an original initiative. Back in April 2020, CWN created the Canadian Coalition on COVID-19 Wastewater-Based Research. And this was exactly that, a coalition of researchers focused mainly on methods development and sort of reflecting the focus of the international community, and was very much focused especially on sample preparation and analysis. It brought together a lot of people from environmental surveillance fields who weren't as familiar maybe with working with public health agencies in Canada, and therefore weren't always as familiar with those sort of typical ethical obligations. And so among researchers, there was a need to establish that the investigation of wastewater for the detection of signals of SARS-CoV-2 was fundamentally an application of public health surveillance and must be governed by the appropriate ethical guidance. And so, as we all know, because wastewater data is potentially sensitive and has the potential to identify groups of individuals, as well as the need to balance individual rights and freedoms with population interests, as Nathalie and Jean-Baptiste got into, based upon the WHO guidelines from 2017 on public health surveillance ethics, CWN developed our own ethics guidelines. And so some of the key takeaways from that, which are still relevant now, but I'll also get into sort of what the current discussions among public health practitioners are in 2024. But from these guidelines, we found that Canada has an obligation to develop appropriate public health surveillance systems and ensure the data collected are timely, of sufficient quality, and developed through validated methods. Investigators have an imperative to work with public health authorities to account for community values and concerns, and to minimize or mitigate risks of harm throughout the life of a program, and especially in vulnerable communities that are experiencing disproportionate harms due to COVID-19. And I'm saying COVID-19 throughout all of these points, but it's really relevant beyond that to other infectious diseases as well. There's also obviously a potential for ill-considered public release of data that could stigmatize communities whose wastewater has been surveyed. And so as a generic default, data should be treated with the same level of security as individual health information, while recognizing that wastewater samples do represent groups of individuals. Data from wastewater surveillance it can provide early warning of localized COVID-19 outbreaks, and fully validated results must be communicated to public health authorities as soon as possible. And this tension between the time needed to validate versus communicate results must be understood and managed by researchers and public health authorities. And that, of course, was, a concern at this time in the pandemic. And so looking sort of post the publication of these guidelines, they're all still very much relevant. They're all still conversations that we're having. But wastewater surveillance in Canada has also begun to move from research initiatives to public health programs, and conversations are moving to other considerations as well in ethics. So in our current peer learning program, among public health practitioners working for the government, who are our key participants, epidemiologists, public health officials known as medical officers of health. And there's three main ethical issues that I'll touch on very, very briefly now, and I'll talk about a bit more in later questions for the panel. But first of all, there are issues of trust, in particular with equity deserving groups, and there is a need to build trust with those communities who have been treated badly by the government, by researchers, by public health historically and currently. And avoiding stigmatization and panic in communities. There's a need to communicate the data, and not just to communicate the data, but to be clear around who has ownership and control over this data. And finally, in this modern world where there's this democratization of data and we have all these accessible ways of information, wastewater surveillance on the sort of other side of ethics provides this really novel opportunity, I think, as we all in this room feel, where people have immediate access to information and data about their health and what the potential of that is. So anyway, I'll leave it there.
Thank you. We'll come back. Sorry, we'll come back to some of the issues later on in the discussion. So thanks for all those insights on the ethical consideration. Let me go back to the legal aspects now. So we understood that there is an issue with mass data collection from wastewater-based surveillance. In the US, for example, do you have any examples of whether it could be used, for example, to identify, let's say, drugs, hotspots, or criminal investigations, or are there some limitations from those key aspects?
Yeah, so that's a great question. So one of the oldest and best established wastewater surveillance programs in the country is in Tempe, Arizona, where they actually spun up their project, I believe, first for monitoring for opioids, in part as a, designed to be a public health measure to identify neighborhoods where they could send additional resources to combat substance use disorder. But of course, as I always like to say, if you build it, law enforcement will eventually show up and want access to it. So I'm not aware, though it would be really hard to find out whether law enforcement in Tempe has sought access to that data. There are other tools that law enforcement might also use for heat mapping opioid or substance use disorder, other substance use disorder. So wastewater surveillance data isn't the only source that they might look to. But it might be a particularly useful one insofar as it can be traced from a really broad population further and further up the wastewater stream to a much smaller population, right? And as we get to smaller populations, that's where my concerns are really heightened about individual identifiability and law enforcement access to those resources. So, what law enforcement does in terms of investigations often isn't always a matter of public record. And sometimes it's obfuscated in the documents that are disclosed in criminal prosecutions. So though I don't know for certain that this has happened, it may have. And opioid surveillance is one of the uses that some wastewater surveillance programs have pursued. Now, I think it's worth noting that the way that US law would treat wastewater surveillance for public health purposes as opposed to for law enforcement purposes can be quite different, right? So insofar as individual level data might be accessible, right? If it's not accessible, then we don't have privacy concerns necessarily, not as a legal matter, that we might have, population privacy, kinds of group privacy interests, right? But that's not really how we think about privacy in the United States. But if there's individual level data that could be derived from the wastewater stream, then its collection and use by the government for public health purposes would be treated very differently and scrutinized by a court very differently from law enforcement's access to that data. We superintend or scrutinize law enforcement access to data more seriously, or at least we're supposed to, than for some non-law enforcement purpose like public health. That's why public health efforts, even when run by the state itself, can collect all sorts of personal data that wouldn't necessarily be as readily available to law enforcement. So I think that's worth noting.
Thank you. Is it the same in Europe, Jean-Baptiste, where we are the GDPR, as you mentioned before?
Concerning Europe, I would like to develop a different example, different situation in order to imagine the possible utilization of WBE data collection. The first one concerning criminal investigation, such an identification of a drug hotspots, for example, but really in criminal investigation, and it is important. Under current law, at least for European law and the mostly European countries' internal law, I guess there don't really exist a legal basis permitting an utilization of wastewater monitoring for criminal investigation purpose. Admittedly, we could imagine find a general legal basis which could be interpreted in order to include such utilization of WBE data collection. However, generally, criminal law is governed by a principle of a strict interpretation. And the example of the identification of a drug hotspot is insightful because we can hesitate in Europe between two legal bases by analogy, garbage search and secret surveillances of data connection. In most legal systems, garbage search during a criminal investigation doesn't be considered as interfering in the right of private life and the protection of the home. But as far as I'm concerned, I'm not sure the comparison with wastewater monitoring is relevant because throw a thing in a garbage is a voluntary act. Unlikely to the bodily excretion. Moreover, wastewater monitoring is more general and global by definition than an isolated research in a garbage. So I think secret surveillance of data connection might be a more relevant legal basis. In this case, a legal framework, excluding GDPR, is really limited. Especially, European courts is very careful about general system of secret surveillance and rules that a generalized processing and storage of a connection data is prohibited in order to criminal investigation. The courts insist especially on the importance of to avoid chilling effects and citizens feeling that all the private life and family life is under a constant monitoring. The prohibition is not absolute, but the legal basis must be very detailed, very precise, and must have a solid guarantees of a protection of privacy life and about transparency. For example, with an authorization of an independent public authorities which control the data collection. So in the case of a drug hotspot, the identification of a drug hotspot, I believe WBE data collection could be used, but not in a primary intention, not in order to initiate criminal prosecution, but also to, but just for only for confirm a point during a criminal investigation. If I have a few minutes, I would like to imagine the case of a very targeted preventive measure to avoid the growing of a new epidemic, for example, in an airport or a port or university. I perceive some limits here about the utilization of WBE data collection. The first one is about the place of the director of the place. For example, director of the school or university, because privacy life, I think, avoid that director of school has health data and sensitive data about the pupils or students. And so I think there is a risk with WBE data collection if a director of school or university has the abilities to detain some sensitive information. And in France, the Constitutional Council had been censored a law during the COVID about this point. Then I think another difficulties is about the risk of discrimination and reputation damages. I'm not certain about that. I'm not very confident, but in the European Court's jurisprudence, the courts insist about the relation between the right to respect for private and family life and the human dignity and individual autonomy, especially the right of self-determination. And so I think about this point, maybe WBE data collection might ask a question, especially if it justify specific measure on a special community, migrant community, or religious, or concerning sexual orientation. But, and if I have only 1 minute, I think the risk depends on the affiliation to the place. And so I think this risk could be limited for the case of the port or airports. But in this case, the most important things is about the key is about the proportionality of the measure. Because I believe if, for example, we would like to avoid takeoff flight, we avoid the takeoff flight because we have information about an epidemic. Proportionality could be a question because of the measure is really strict regarding to in relating to the European Court's case law. Thank you.
Well, thank you. So you raised some issues of ethics, right? I mean, can be for the use of opioids, for example, or even for the levels of infectious disease that you can find in some communities. How do you deal with that concretely? How can you minimize this kind of ethical risk vis-a-vis, especially some communities that are already, that might be already stigmatized?
Yeah, so when I was thinking of the best example to illustrate an answer to this question, I kept coming back to this underlying idea of, the best way to minimize ethical risk is by partnering with the affected community for a very, at least community informed, if not community-led response. So a really great example from the province of Ontario in Canada and from our program partner was through MPOX wastewater monitoring back in 2022 from our program partner, Adam Awad, who at the time was the communications manager for a community organization, the Gay Men's Sexual Health Alliance, or GMSH. And so as news of MPOX started circulating in spring 2022, the GMSH developed a community advisory, and this was one of the first in the country. And this linked community members to reliable information about the virus. And as case counts started to rise, provincial public health authorities quickly identified the GMSH as a key community partner in the response, especially leading up to Pride in Toronto. And in particular, with MPOX being monitored in the wastewater, there was a need to discuss the communication of potentially sensitive data and what those impacts would be on this particular equity-deserving population. And, I think as we all know, there were a lot of uncomfortable parallels to early days of the HIV pandemic. And there's this level of mistrust between LGBT+ community and the government and healthcare. And so the GMSH were able to coordinate regular meetings between community-based organizations, public health authorities, researchers, and policymakers, which helped ensure timely feedback and collaboration between everybody. And there was also other campaigns, including a public education campaign led by GMSH, which targeted LGBTQ+ men, including a video by a drag queen and describing in graphic detail how people can avoid being infected with MPOX. And so, this sort of cross-sector coordination and really innovation in integrating with wastewater surveillance, it improved the speed, the quality of the response, and it made Ontario's response to MPOX one of the most effective MPOX responses in the world. And this experience in Ontario really demonstrates how we can minimize stigma and this sort of ethical risk, and shows the importance of leadership, engagement with communities, and it provides a great example of this sort of integration within integrating all those activities into wastewater monitoring. And I think it's sort of in contrast to other community organizations that we've spoken with, particularly those concerned with, drug surveillance, drug-involved communities, and the impact of the history of those communities being so heavily surveyed. And so, as Natalie sort of mentioned earlier, if you build it, law enforcement will want access. And so a lot of those concerns were also echoed by Dutch researchers who we spoke to. And so this sort of mistrust, and when we think of community organizations, we need to involve them, we need to engage and incorporate that into our wastewater monitoring and focus on the sort of public health action and not just the possibilities of wastewater monitoring.
Just a small question on that. So you talk about basically involving communities, communicating with them, but do you have like a moral obligation to say that, for example, you found some disease? Thinking about mpox, assume that you do it in my university and that you realize that there's a lot of mpox. So how do you communicate on that, for example?
Well, so I think with a moral obligation, I think the bottom line when I look to CWN's ethical guidance and others, the bottom line is there is that moral obligation. I think when you're collecting that data, you have an obligation to communicate it back to the affected communities, right? And I mean, maybe I'm jumping the gun with another later question, but in Canada, a lot of these discussions around ethics of wastewater surveillance came from public health practitioners serving First Nations, Inuit, and Métis communities. And so there's lots of ideas there and a need for Indigenous sovereignty after a long history and current reality of colonization in Canada. And the need to, when it comes to moral obligation, follow principles such as OCAP, which stands for ownership, control, access, and possession. And so, yeah, that's a part of the moral obligation aspect. I'll maybe leave it. I don't know if we'll return to it later, but there's those sorts of principles and others. That's really what we want to consider with that sort of moral obligation.
All right. Natalie, you want to react?
I mean, I think that there's space between the kind of minima that law establishes and the command of ethical theory, right? So there's daylight there that, US law doesn't prescribe, doesn't require nor prohibit all that much in this space, except really on the margins, right? But ethical practice can impose a much more, a much better guiding framework, right? So public health only works when we have public trust. And so public trust is a necessary predicate for successful monitoring, implementation, uptake of community responses, right? Wastewater surveillance gives us a lot of raw data, and we might not need people's consent to get it, right? But if you want to use it ethically, as we heard earlier this morning, you've got to have a plan for what to do with that data. At what point does the viral load indicate that a particular public health intervention is warranted and ethically obligatory? And if you go to the community and say, oh, well, we've been spying on your wastewater all this time, surprise, now get a vaccine, that's not going to go very well, right? You have to have buy-in from the community, acceptance from the community, support from the community, right? And that only becomes more and more important as maybe the watersheds get smaller or less community-wide, right? Because then they can become very population-specific, right? And subpopulation specific and disadvantaged group specific, right? And that's where we see, I mean, in the United States, as in many other places, there are historically disadvantaged and marginalized populations. They have a lot of distrust, rightfully so, with both the medical and public health and other communities and scientific communities because of a long history of mistreatment or lack of attention, right? Lack of attention and not the kind of attention you want, right? And so it really requires extra work to bring people along And that's hard work, but it's absolutely necessary if wastewater-based epidemiology, wastewater surveillance is to serve the public function it says it's in pursuit of, right? If it is in fact for public health. Now, if you all want to go collect all your data and get your fancy publication, maybe all you're doing is research, and maybe you don't care about interventions into the community, for which I would say, that's too bad. I think also you probably owe something to the populations you're involving in your research. But, you know, the requirements of research and the requirements of public health should are connected. They may be overlapping, particularly in this space, right? And so I think there's a lot more to say about what ethics suggests than what law can prohibit or require around the margins. Does that make sense?
Thank you. Okay, so I think actually we are almost, we have 10 minutes for questions. So if there are any questions for the room. Anybody? Yeah. Should I give you the mic or? Yeah. Thank you.
Okay, thank you. Hello, thank you for your intervention. Just wanted to make a comment upon all of that you said. As Natalie said in the first place, people contribute to the data we collect, like they actually shred all their things in the toilets. And if we actually use this data against them, we won't have their trust and they won't collaborate and they will find ways to don't collaborate to this is my it's actually something we're not thinking about. Like people, when they get offended by a system, they try to avoid it and they try to create their own system. So we just need to collectively think about how they can trust the system and not work against it. That's just all I need to say. And thank you for everything you've said.
Yes, hello, Frank Ostrup from Denmark. Absolutely not my area of expertise, but super, super exciting. And I think it really also important to have this discussion. I don't know, I was just feeling a little bit that most of the discussion was, you know, on the rights of the individuals, you know, that are infected or might be, can we detect them? But I just wondering, and it's probably just out of ignorance, but what about the rights of those that are at risk? You know, we also need to have some kind of a system in place to protect those that are at risk of becoming infected. So where is the balance there? And I don't know if you could just reflect a little bit on this, that yes, we should protect individuals, but we should also protect the population. And I know that Hippocratic, you know, oath starts with the population and then, you know, individuals. So at least medical doctors know that's what they should think about, the population and not the individuals. But just a bit of reflections there. Thank you.
So I think one way to think about this is to think about whether there are tools, legal or otherwise, that might be interposed between the data collection and the things that we worry about, so that we can reap the benefits of wastewater-based epidemiology for the community without running the risk of undermining that success by scaring people or pushing people out of the system, either from their own concern or because we're using their data against them, right? So here I think about a tool which I haven't had a chance to mention, but which is really quite an interesting one in US law. When someone is engaged in research and that research is using or developing sensitive and identifiable information about an individual, then if they are funded by the federal government, they are entitled to what's called a certificate of confidentiality. And that's a special legal protection that allows them to say to anyone who wants to compel access to their research data or the data about their research participants, no. In fact, it's an obligation. You must not disclose that data, right? And that applies really strongly even against law enforcement, right? So if we're thinking about that as the privacy harm, right? Then at least for research, developing or using information about identifiable humans in the United States for federally funded researchers and non-federally funded researchers who ask the federal government to give them this protection, a certificate, they have a really great tool to pursue the benefits of that research for the population at large, while being able to represent to the communities they're working with and the participants in the research, whether with informed consent or frankly, even without informed consent, they can represent your data is secure. I can protect it even if someone comes to me with a legal court order that says you've got to give up the data. That should not, that shouldn't open up the research. Even a court order should not open up the research to public view, that individual level data. So I think that's one tool, right? That exists for research. It doesn't exist for public health data. Maybe it should. And it's a US specific protection. And what US law enforcement can get access to from US based researchers says nothing about what European police can get access to from European based researchers. So I recommend to you the certificate of confidentiality is a great model for reaping the benefits without imperiling trust.
If I understand your question is about the frontier and the place of public population protection and individual privacy. In fact, in Europe, there is no doubt on a point. States has a positive obligation to protect health under the Article 8 of the right of privacy and private life. So the question is about the balance between individual rights and the protection of population, and the concept of proportionality and public interests and the necessity of data collection, I think, are about precisely this balance. I think WBE data collection is a really difficult thing for an European lawyer today, because we have not precedent case law to measure the proportionality. And also, I think there is a difficulty which come from the conception of privacy, because privacy is, and the GDPR the same, is linked to the natural person, to individual person, and not about the group. And for WBE data collection, I think there is a specific question about the discrimination of groups. And in Europe, I think we have not juridical concept to think about that.
Sure. I'm so sorry. I'm going to jump back in because I think it's a really good question. And I just wanted to note that one other way to approach this question is to think about what practices can we put in place that maximize public benefit and minimize the likelihood of individual identifiability, individual privacy being imperiled, right? So that would say we should have minimum numbers of the number of people that should be in the watershed that we're sampling. We should think really, really carefully about whether to shield or include in our data dashboards cryptic lineages, unusual lineages that might be traceable to a single individual. We should think really carefully, all you folks into metagenomics, about whether metagenomics exacerbates privacy concerns because it develops all the data and researchers want to store all the data forever and all the samples forever. And privacy law says we should minimize the data we're developing and we should destroy it as soon as we don't need it anymore, right? Now, those two are intention, absolutely. But that should be what we're thinking about. How can we minimize the risk of going too far up the wastewater stream, right? And yield the maximum benefits of wastewater-based epidemiology without getting that individual identifiability.
I would like to precise the last point and come back about the idea of public trust, because I think the question is not to know if we can find a legal basis or legal framework about WBE data collection in Europe. Juridically, we can justify everything, but that's not the point. The point is really for European law, the ethical question and the public trust pass by the law, and the law must be clear and predictable. And if the utilization is really clear, if the protection, the guarantees is really clear, so it's okay for the right to privacy. But we need to legal basis really clear and really predictable for the people.
Any other questions? Yes. Thank you.
Good afternoon. Thank you for your presentations. I would go in the same line as Frank. One thing is research, yes? The other thing is public health. And so the governments, they have the duties to serve the population and protect the population. So for example, we have eliminated measles. And then in the wastewater, my wastewater samples that you have thousands, millions of people there, I find a positive sample. It's my obligation to follow up, and I have to reach the individual. Otherwise, I will put the community under risk. It's a threat. So I'm very sorry, but I cannot understand that the only state that by constitution must protect citizen is not doing his job because I can hurt somehow the individual privacy. And so I agree that we must be transparent. It's very important. I agree that we need the community engagement because it comes from bottom to up, and then it will reach the governance, the high level governance. But despite of all of that, how are we going to sleep as public health professionals, knowing that we have found, we have communicated, the Ministry of Health is looking at that, and we do nothing because of privacy. And so this is a total imbalance. And I would like to listen to you. That's it. One thing is research. Okay, then I don't have the rules. I don't have duties, legal duties. The other thing is, as a government, it's omission. It's help omission, which is a crime, at least in Brazil, and I think in other countries as well. So, but I'm not a lawyer, yeah. And they, even my knowledge on ethics, it's far from yours. So I would like to listen. What about a public health approach? Thank you.
You have 90 seconds. Good luck.
If I understand your question, it is about the responsibilities of governments who have an information and it does not act. I think, and I'm sure that a government which has an information, important information about an epidemic, for example, and which doesn't act, involve his own responsibilities. For example, in France, it is the case. But the question is really difficult to establish, legally speaking, because of the question of proportionality. And proportionality is not only about individual privacy, but also about economic interests, about religious interests, about other rights of the human. And for example, if I take again the example of the airplane, the question could be about the potentiality and the reality of the risk. If, for example, WBE data collection permits to identify only a potential risk with 1 or 2 persons and really early, maybe stop the airplane could be appreciated as a disproportionate measure. Whereas, for example, if WBE data collection identify that all the passengers are contaminated, stop the plane could be proportionate. I don't know, but legally speaking, maybe it could be the good reasonment about it. I don't know if I answered to your question.
Natalie wants to react.
So as I said earlier, even when we have individual level privacy interests, the intent of the party of the government entity doing or making use of that information can really shape how the law thinks about that use. So the public health entities are entitled to collect information that law enforcement wouldn't be entitled to collect without a warrant, right? But public health entities can do it population-wide. The really difficult part comes when law enforcement wants to tap public health's resources and make secondary use of that, right? Or turn public health into public surveillance for law enforcement purposes. And so I think what's really essential here is if we're going to continue to believe, as I think we probably can, that if there is a measles case, we'd better identify that measles case and their close contacts, because and basically anyone they came in contact with, because measles is so infectious, right? That we'd better put really strong protections in place around not only the data about that individual, but all the data that gets funneled into that measles surveillance program, so that it doesn't get redirected to a use that really conflicts with public trust.
Yeah, one last question.
Thank you. I think that the problem does not only concern the relationships between individuals and the population, but also groups or communities, as we said again. But it's a real concern that a community or a group should be designed as responsible for an epidemic, for a pandemic. And we've got some kind, well, this with about AIDS and the homosexuals. And it can be the same with refugees, with migrants, simple foreigners. So how do you cope with this risk?
I mean, I can speak on this a little bit, and I think I just go back to that example that I spoke on from MPOX. And the key really here is to have community-led or at least community-informed responses. And when we're worried about this backlash on these communities who are already vulnerable, who are already marginalized, we need to think about how to have them to have an equal seat at the table, working with policymakers, working with public health. I think that's the key way that we can move forward.
Look, law doesn't have a really good answer when it comes to group identity, group privacy interests. Privacy law, at least in the United States, and perhaps it's more relational in Europe, but in the United States, privacy law considers every man as an island, right? We're all individual atoms, as if we were born out of the ether and have never, and don't rely on anybody else, right? So relational privacy and community-based privacy is something that we just don't know how to think about at least in American law. It is something we are beginning to realize we really have to grapple with sometimes. And again, this comes up most prominently where research interfaces with Native American groups, Native American communities, indigenous communities, right? Because they do sometimes, some tribal communities have a really robust sense of community identity, community privacy, relational privacy. And so trying to accommodate that within the scope of research has been an ongoing project. You know, it's a work in progress, and it's something that is not articulated in law as much as in ethical practice.
It is really difficult to speak and to think about the groups for an European lawyer, and more especially, I think, for a French lawyer, because we have a really specific philosophical conception of the group. But I am agree with that, and I think the point is really privacy is conceived about and specifically about the individual. European courts can conceive the group, but with the relationship between individuals and the right to have mutual relation with others. And for example, in WBE data collection, if we think about a very targeted measure, stigmatizing a neighborhood, for example, we can imagine that European courts says, okay, but there is a risk to lead some relationship with the other. But I'm not sure about that, because privacy is really about the individual in first.
Thank you so much. So it's time for the coffee break. We can keep discussing, continue the discussion at the coffee break. Thank you so much, Talia, Natalia, and Jean-Baptiste, for the insights. Thank you.
And attendees. I am very honored to introduce Dr. Chekwe Hiekwezu to you today as the keynote speakers of today's International Conference on Wastewater-Based Surveillance and of Infectious Disease. which is organized by the 4U+ Alliance, Sorbonne University, GIS OBEPINE, the Geneva Health Forum, and Panthéon-Assas University, with the support and participation of the World Health Organization. Dr. Iekwezu's expertise and leadership in public health and infectious disease surveillance make him a prominent figure in the global effort to enhance health emergency preparedness. Before giving him the floor for this keynote in our conference today, allow me to briefly share some of his background. Dr. Iekwezu currently serves as the Assistant Director General for the WHO Division of Information, Surveillance and Health Emergency Systems, where he oversees the WHO Hub for Pandemic and Epidemic Intelligence. In this role, he collaborates with partners worldwide to anticipate and mitigate pandemic risks through improved data access, analytical capabilities, and decision-making tools. Previously, he was the first Director General of the Nigeria Center for Disease Control, where he served from 2016 to 2021. Dr. Iekwezu holds a medical degree with a specialization in infectious disease epidemiology and a postgraduate degree in public health. It's a privilege to have him with us today, and without further ado, I'm pleased to hand over the stage to Dr. Iekwezu for his keynote address entitled Wastewater Surveillance, a Global Perspective. The floor is yours.
Thank you, and good evening, colleagues. Real pleasure to be here. It's very rare these days that I have the opportunity of thinking about a topic, as most of my time, unfortunately, is spent either fighting existing fires, trying to get countries to agree on a new pandemic treaty, or just raising money in a very difficult geopolitical landscape at the moment for WHO. So I've actually enjoyed the hours I've spent thinking about what to say to a group of experts that obviously know a lot more about this topic than I do, and rightly so. But I think it's an exciting time for this because, as we reflect on the last few years, going through the pandemic, now coming out of it, new methods emerging, being merged into public health, all the interesting questions that are being asked. It's just really exciting to be part of that process and to hopefully contribute to it one way or the other. And I just wanted to start with this obviously extremely simplified image of some of the challenges that we face at the moment and really focus on this term, uncertainty, and how we deal with it. I framed it in the context of a pandemic, but you can put epidemic there or outbreak or however you want to define it. And ultimately, this is what most of us do, trying to make sense of uncertainty, and whether we're doing it from a microbiologist point of view, an epidemiologist, or any of the sciences that drive our work. And this really, if I think of any of the ongoing incidents that we're dealing with at the moment, we would be somewhere along that curve. Today I go home, we'll be talking about H5N1 in the US. We're thinking about whether it's a true outbreak. There will be a briefing on Marburg in Rwanda. We'll be talking about containment. There'll be an update on Mpox and the response in DRC and neighboring countries where we've just agreed a vaccine allocation to a number of countries, depending on several factors. And for none of these decisions are we actually ever certain that we're making the right ones. We make the best possible decision with the best possible set of bits of information that we have. But this is the uncertainty. Every morning in WHO, we sit down at 9 a.m. and we think through what are the new pathogens, new threats that have been reported the day before, where have they happened, what's the severity of those affected, should we respond, should we not respond, should we allocate resources? And it's the same thing that I'm sure happens at the national level, where I've worked most of my career before joining WHO, where many of you work. So this is the context in which we work. And unfortunately, it seems to be getting worse. So any of us in this field, I can't, hands on my heart, say, listen, life will get any easier for us. And some of it is completely out of our hands, but certainly a lot of it is, we humans driving this. And in WHO, we really don't have the option of choosing what to respond to or not, because these things keep happening. Some of them are just the health consequences of either humanitarian issues or conflict or emerging or old infections like cholera that last year, you wouldn't even imagine the number of people affected by this very old disease. And we still have to respond to all of this. So at the moment, we're dealing with 41 graded health emergencies globally. So there are 41 health emergencies where countries expect WHO and where we are deploying extra resources to. So that doesn't include any of the incidents where countries are managing themselves to respond to. So our work is cut out for us as a global community, and that's important to place wastewater surveillance in that context. And some of these things I'm sure you'd have talked about ad infinitum since this morning on the value and for what and for whom, and we'll talk about some of them. And in some cases, it's easier than others. With polio, it's an old example. I'm Nigerian, so we've had this problem for many years. It's not a theoretical construct for me personally. We're still battling with this because every day, while wild polio has been eliminated in Nigeria, we still have a lot of the vaccine-derived polioviruses being reported from time to time. So this is a big challenge. It sounds, even though polio is probably the best use case for wastewater surveillance, in of itself is still extremely difficult to do. Extremely difficult to choose what sites, when, how, with whom, what samples to collect. What happens when there have been floods 2 months ago that almost devastated the northeast of Nigeria. So what impact does that have on many established sites? How do you sustain the laboratory infrastructure and testing for this? And I'm using this as polio, for which, is the strongest use case for the thing that we're talking about today. So this is not an easy problem that you have chosen to think about. We have chosen to think about together. So for each of these, the emerging evidence, and probably the evidence is weaker as you get down and even weaker as you start going further down for the new pathogens for which we can think about. But it can't stop us from pushing on this new opportunity to enhance public health surveillance. And that's why we've gathered here. And, and I kind of want to focus us a little bit on these three questions, which is really what we're trying to answer. And this is not where the questions will end, but I think if we make sense of these three, then we can start digging forward and answering others and expanding the set of questions that we seek to answer. And I'll make the case at the end why that none of these questions can be answered by any of us in this room on our own. And ultimately, we will have to find a way, a model of thinking and working together in order to make sense of these. So what pathogens and what settings can wastewater surveillance be used? What are the evidence gaps? Sometimes I feel we're moving too fast ahead without answering the second question specifically. And then what are the global needs? What are the global goods needed to enable member states to integrate wastewater surveillance into a broader platform of surveillance opportunities in order to enhance decision making? But before we go into the specifics, let me try and frame this into that global framework in how we work, because ultimately our responsibility is around emergency preparedness, prevention, preparedness, response, and resilience. That's how we define what the World Health Organization's emergencies program does. It's really around those 4 terms. And how we've defined this is around what we call the HEPA framework. So if you look at the vertical bars there, you find pandemic and epidemic threats, humanitarian crisis, and all the other threats. So if you that's a threat perspective going from up to down. And the capabilities that around which we organize how we work really is arranged around these 5 capabilities. So on top of that, you see collaborative surveillance, and I'll go into that. But before I do that, above that is the governance that is being spoken about at the moment. Like I said, we're discussing a new pandemic agreement, and we just passed new international health regulations a few months ago. And on the other side is all the financial instruments that we need to do all this work that we have to do. So for anything we're doing, we need context, we need a framework. So this is the framework of the work we do all together. Now, if we pick up just the collaborative surveillance and dig into that a little bit further, we've defined it as the systematic strengthening of capacity and collaboration. And I'll keep coming back to this word among diverse stakeholders, both within and outside the health sector, with the ultimate goal of enhancing public health intelligence and decision-making. And we'll use this term a lot in my next few minutes, decision-making, because if we're not getting to decisions being made with surveillance, then we're not doing surveillance. And so we have to really think about this word, decision-making, and how our efforts are leading to decision-making. And the way we think about it is around these three strands, and then getting to the image at the end. And you would wonder why we have that image of a rope, because ultimately the point we're trying to make is that unless we tie this rope together and get it to be as strong as we imagine it to be, that every rope is stronger significantly than the strands of each of them. So think about surveillance as coming together, collaborative surveillance, both around disease threats between the different sectors, around emergency cycles, and collaboration across geographical levels. And to do this effectively is our best chance at getting to the outgo that we want, to the goals that we want. So this is an image that we try to use to describe what we're calling collaborative surveillance and trying to position wastewater surveillance within that. Now, anyone that has worked, is working, has trained in surveillance, sees it from different aspects, and we've all learned the traditional definitions, and we all put a lot of effort into this. And we will find ourselves in one of these boxes, whether we're doing surveillance on the health sector, in the One Health, any of the sectors, whether it's verticalized disease programs or integrated programs, whether we're looking at mortality in hospitals or at countries, we will find ourselves in one of these boxes. But if there's no learning out of this pandemic that we have, is that the traditional notion of surveillance, of counting incident cases and maybe deaths and a bit of morbidity, is just simply insufficient for our leaders to make the decisions that they have to make. So if we think of whatever country we're in, or we're from, or we were in, I led the response to one of the biggest countries in Africa. And previously, when you would ask us to provide, if my minister asked me to provide him data, I can tell him, sir, I'll go back, do some analysis, and come back to you in 2 months with a few graphs. The pandemic showed us that that answer was simply not acceptable anymore. You had to face the country almost every night and then every week and explain to them what was happening in the past, what you projected to happen next week, what will happen in 2 months, when will we get the vaccines, what impact will it have, how many people will be in hospitals, how many ventilators do you have enough oxygen? And, you know, surveillance as we had learned it was simply not sufficient to answer those questions. It was not. And even in the most advanced countries in the world, yes, some of them got better, 2 months, 3 months, sometimes 1 year into the pandemic. But in those first 3 months, there was no country, hand on heart, that can say, listen, I was able to provide my prime minister the data they needed to address the nation that the next evening on what was going on. So we had to think about how do we start bringing this together. And so wastewater surveillance has really emerged as an opportunity out of that. But I think that the anxiety and opportunity has almost driven us too far ahead without doing the basics. And these are just a few of the examples. And for each of this website and for each of this country, I've asked what decisions are being made from the data being collected. We are not getting the answers on the public health decisions being made on the investments in public health, in wastewater surveillance. If there are answers in this room, I'd love to hear them, but I haven't gotten them. So the question is, why are we not making the consequential decisions that we have to make out of the investments that are already being made? So countries are investing a lot of money in all these systems. So do we need to take a few steps back and do a few things and ask a few questions? Have we, and we take some of that responsibility as WHO, have we been too slow in doing what the world expects us to do, in organizing ourselves to arrange the guidelines, the norms, the standards, the opportunities to do things in a standardized way? Have all of us as a global community been too slow in responding to the opportunity that wastewater provides? And what is the consequence of this? And I know we've all used this image in different ways, but in a way, I feel that's exactly what is happening. We're finding what we look for, or sometimes not finding what we don't look for, and we're making decisions on something that we don't know how it relates to the decision everyone else is making. It reminds me a little bit about the beginning of genomics as a public health tool, and the fallacy of imagining that a genome can tell us anything without being able to be compared to everything else. So in a way, that's kind of where we are. And until we define those parameters that we can compare these to each other and to an expected level in a population, we will not be able to make the best out of this. And that's the realization we've come to, and set these 5 priorities for ourselves within WHO to start organizing this space and to take a few steps back and say, how can we drive these 5 important areas of work and hopefully help us build this together as a community? So in our initial work on the global landscape analysis, many of you in this room would have been contacted. If not, you'll be contacted in the next few weeks to understand what countries are doing. Those 5 countries I put as examples, there are many more. How, learning from colleagues that have invested time, energy, research capital, research resources on answering these questions, and beginning to make sense of this. What are the opportunities, the best practices, the challenges, the funding mechanisms? How are you integrating this into your national public health systems at the moment? Is it being done completely in parallel to all the other surveillance approaches, or is it being done with and inclusive of all the other national public health surveillance systems in the same country? So how is this being run? And what are the challenges that you are finding? What are the standards we need to work towards on data collection, on sample collection, on transportation, on lab testing, on visualization? All important questions. And a lot of work has been done on this. It's just that it's been done differently, implemented differently, and challenging us. Of course, I'm speaking from a global perspective. And so if you need to make sense of what is happening globally, you need to have a way of comparing X with Y. And if we're not able to do that, then we are really challenged. So data standards, the very important one. And of course, data in a way is not just about the data, it's all the mechanisms involved in everything needed to generate the data, visualize the data, analyze the data, and everything in the data ecosystem. Very important ethical questions that we haven't started really answering around opportunities, benefits, and what are the trade-offs. And these are so important. And if we don't do it in the beginning, we will end up in a challenging situation. And you can imagine all the scenarios that are possible on this, on what samples collected are used for, and for what benefits, and for whom, and how do we ensure that communities from which samples are collected also derive some benefits from this? How do we build the transparency, the trust in communities from which we are collecting samples? What are potential harms? Have we thought about it? Who owns the data? One thing is having the opportunity to display the data on a dashboard, but who actually owns the data? And what are we allowed and not allowed? And I just came in at the last part of the conversation in the last session. So very important questions that we will not serve ourselves well as a community if we don't think about them, put them on the table now, answer them as best we can, and keep improving the answers as we move along. How do we develop basic guidance and guidelines so that countries thinking about doing this don't all have to do this from scratch? So even if the guidance is not perfect in the beginning, at least do we have a standard from which to start off? So if Togo requests WHO to offer them, how do we start this? So that every small country in East, West, North, and South doesn't go through the same startup process of thinking, how do I collect samples? How do I identify labs? Where do I send them to? What methods do I use? And everything else related to these important questions. And finally, this is an opportunity. Lately, this came out of the genomics work, actually, and the emergence of genomics as an important tool for public health. And so we curated a new international pathogen surveillance network trying to almost solve very similar problems that the wastewater community has now, but bringing together a network of genomics actors. And they are now thinking very hard together on a lot of things to define the work they do, bringing together the different communities doing this, defining communities of practice around specific things, thinking about how to scale up, how to maximize the opportunity that funding has offered this space. Genomics has probably received more funds than any other aspect of surveillance post-pandemic. And if you look around the developing world today, you will find a lot of the equipment that has been purchased not being used because the equipment costs, as high as they are, will not solve this problem without the expertise and the knowledge and the community that needs to drive this opportunity forward. So we're about to make very similar mistakes with wastewater if we don't think about these things. So one of the most active groups in IPSN is a committee of practice on wastewater and environmental surveillance, trying to bring these two exciting fields together to think about the feasibility of using wastewater for in the outbreak context and all the capacity development challenges listed below. So in the midst of these challenges, there are also a number of emerging opportunities. And the exciting thing is that they're being driven by colleagues working in this space. And I'm really excited about some of the emergent opportunities. So to round up, these are things I think we need to focus on. And these are real opportunities. I'm excited, as you are, about the opportunity of wastewater surveillance. I think there's exciting work happening in many countries. Nothing that I've said here would be a surprise to anybody in the field. I'm sure you're thinking about the same challenges, whether from an academic perspective, a national public health perspective, a funder's perspective. I speak to many of the funders. They ask me, who do I fund for what? The answers are not obvious because we are all asking ourselves the same questions. So I do think there's an opportunity for us to come together. And when I say us, I am very humble in including WHO in that we have to really work together, think together, and allow ourselves some humility and some vulnerability in accepting that none of us has all the answers. And we have to really think very hard on how to solve these problems, how to integrate this incredible opportunity into a public health surveillance landscape. And not think about it as a standalone opportunity, and really think about it from a decision-making perspective. How does the data that we collect add to the opportunity of making better decisions for the people that we serve? How do we collect the data, share, and collaborate based on the data and the trust that is required to do so? And when I speak about trust, it's not just trust between countries, it's trust between communities, between vulnerable communities and their governments. And all the opportunities that wastewater provides, inherent in all of that is a risk, because in all of that, we tend to assume that we have an opportunity of skipping something that has been important in the rest of the surveillance landscape of taking samples from humans and the responsibility that comes alongside that, spoken or unspoken. And then the real challenge and opportunity of climate change. So climate change, of course, is a huge challenge, but it amplifies again the speed, the urgency that is required for us to do this work and to think about this beyond the biological hazards that we can answer. So I put up these 4 concluding statements of an opportunity to expand the current scope of wastewater surveillance, and really want to end by appealing to all of us to think about this with a certain level of humility as we move forward, to say, listen, there are more unanswered questions than there are answers. And to answer each of this requires all of us to give a little bit, but to also be open to contribute to a much bigger set of goals and really start today. And that's why I'm really excited about this, the gathering here. I know the work that has gone into it, almost a year of planning, and that we've contributed to a little bit, maybe more, and hope that out of this we can activate a community that will grow the science and grow the opportunity to answer some of the very difficult questions that lie ahead of us. So thank you very much.
Please have a seat. Thank you. I welcome Antoine Flao, my co-chair for this session. Antoine Flao is the director of the Global Health Institute in Geneva. Welcome, Antoine.
Yes, thank you. Welcome, all of you. Thank you, Chikwe, for this very inspiring talk. And maybe we may take a couple of questions from the audience before we start the panel. So I don't know if you have any specific question to Dr. Ekwesu. Yes, please.
Hi, I have an online question from Katharina Helmke. She's asking, is IPSN an additional organization to GLOW-ECON, as GLOW-ECON is also bringing global actors together?
I can speak for IPSN. It's the International Pathogen Surveillance Network. It's a network of genomics actors, so actors in the genomics space, which really grew out of the pandemic, where we saw a lot of the drivers and leaders of genomics for public health were not in the traditional set of surveillance actors. So a lot of the academic institutions that were driving this work, the funders. So IPSN has brought them all together. Within that, there's a subgroup that is interested in wastewater and in terms of how genomics integrates with wastewater surveillance. So there's a community of practice, there are communities of practice around different things. So that's what IPSN is. I think it's slightly different, has slightly different objectives from GLOW-ECON.
Maybe I have one question. Already this morning, some speakers showed us that wastewater surveillance can show us very early some results and can be predictive. But what about the decision making? And you also mentioned that. And I was wondering how can WHO can help us for reinforcing the trust between the politics or the health authorities, between the scientific world and the health authorities to act after understanding what's going on.
Yeah, it's a very important question. And I don't doubt that there's a lot of work that has been done on the opportunity to predict events, predict the scale of events, predict all sorts of opportunities for different pathogens using wastewater surveillance. There's a lot of evidence on that already. How that translates to decisions is a much bigger challenge. And sometimes the decisions we have to understand, most public health decisions are also political decisions in terms of what to do about it. Whether you think about, you can measure lots of things in wastewater, many of them from a public health perspective, uniquely considered, we would think useful interventions. But we have to think about the populations affected and the impact it has on them. If you think about Mpox, let's take a current example that we're dealing with. There are opportunities emerging on the use of wastewater surveillance for Mpox. But if we think about the populations that are vulnerable to this uniquely. And in many countries, there are lots of social, legal implications to this population. The question then is our surveillance activity leading to harm to affected populations? And what responsibility do we have? I don't have all the answers, but I think these are important questions to consider as we think about the benefits and the use of wastewater for decision making. Antoine.
Yes, I may have a question. If you have any question, feel free to raise your hand, of course. Yes, please. So, no, I will after you.
Yeah, thank you for an interesting presentation. One of the biggest challenges we face with implementation of wastewater surveillance, and I think surveillance in general that extends to clinical surveillance, is about supply chains, particularly in low and middle income countries. Is that something WHO can play a role in thinking about sort of, you know, like with essential medicines, you know, essential components to the tools needed for surveillance, or is it just down to member states to have their own kind of battles with companies and suppliers?
No, I think the answer is somewhere in between. You know, when you think about WHO, as you know, we don't really have any hammer or stick to force anyone to do anything, but there are lots of tools and ways that we do that. If I think about a real life example at the moment, we're convening countries, suppliers, and recipient countries on vaccines for Mpox, right? We know at the moment we needed to do that, and we've defined a vaccine allocation mechanism for this round. But we also know that there's no way in the world this can continue at the current price of vaccines for Mpox. So you do that in the short term to respond to that. But then you also convene countries, you convene companies, and you start negotiating and finding a way to both reduce prices or define interesting pricing, differential pricing mechanisms, and all the interesting tools available to us. So I do think in the long term, there's an opportunity to do exactly that. It's not unique for wastewater. Maybe it amplifies the challenge, but I think it's a big challenge, especially in the diagnostic space. I think for vaccines, there have been methods in a few therapeutics, like for the big diseases like HIV, malaria, there have been solutions found. But I think the diagnostic ecosystem is probably the worst served for appropriate pricing of the tools that they need to do this work.
Yes, so I wanted to say that you are the one in WHO, or among the one who help converging all sources of data. And you have presented a slide where you showed the need for integrated data. And this morning, Maria de Roda Hoesman from the Netherlands said that integration of results with other surveillance data might be tedious. We have thought about that this morning, and some people say that there are some reluctance from public health agencies, sometimes from public health professionals themselves, to adopt and to integrate these data from wastewater surveillance. What is your opinion regarding that, and how to overcome and to try to solve this issue?
I wish I could, but first to say it's not unique to wastewater surveillance. And this is a big challenge for all of us in this community, because there's something about public health data that seems to lead to a mental block. And I almost think about this. The only way I can think about this is to look for examples outside of health. And to imagine that there are many other sectors that, you know, have solved this problem in different other ways, right? The fact that our financial, we tend to think that our central banks are somehow unique institutions of national sovereignty, and that is true to some extent. But the amount of data shared by the financial sector globally is incredible. And there will not be a financial sector globally if this data wasn't shared within a framework that allows the sector to work. The reason why airplanes are not bumping into each other is because the aviation industry has found a safe way to share data without compromising their national sovereignty, their national airlines, the prides of each individual country. But somehow, we haven't done that in public health, and we haven't pushed sufficiently and illustrated this as a challenge that affects all of us. And when we talk about data sharing, very often people think about the lack of sharing between countries that are somehow adversarial to each other. We just happen to be in Paris. I would challenge colleagues to think how much they think public health data is shared between Germany and France, two of the biggest allies in Europe. Very little. And it's not unique to Paris, it's not unique to France and Germany. I could say the same between Nigeria and Togo. I could say the same between the US and Canada. So the friendliest countries in the world haven't found a way to solve this problem for the oldest data streams that we know in public health. So I think let's not package it as a wastewater surveillance challenge, but rather as a bigger challenge for all of us working in public health surveillance and how we can, make better sense of this. It was solved, we all followed the example of how genomics data was shared. It wasn't through any big pact. Lots of it was shared on a single platform built by a single group, which ended up being vulnerable for the same reasons that made it so attractive in the first place. So this is a big challenge for all of us to solve.
Thank you so much. So now we will go to our next speaker.
So roundtable, yeah.
Roundtable.
So we may invite you to come to the stage. So starting with Ece Oselik, you are a health policy analyst at OECD, and we will listen to you in a few minutes.
Then Maria de Lourdes Aguirre Oliveira from the Fiocruz, Brazil, that we already had this morning as a chair of one session.
And finally, Wolfgang Philipp, you are principal advisor and chief scientist officer at HERA. You will tell us what is HERA at the European Commission. So we may start with you, Ece. You wanted to present an approach in modeling wastewater surveillance in the field of infectious disease in OECD countries in the context of the pandemic preparedness. So the floor is yours.
Thank you very much. Hi, everybody. My name is Ece Özçelik. I'm a health economist at the OECD's public health team, focusing specifically on communicable diseases. And a big part of our work is actually on pandemic preparedness and response, and what can we do to make an economic case to convince countries to invest in pandemic preparedness. So we currently have a project that's ongoing. And again, our main purpose is to be able to show to the countries of not ministries of health only, but also to ministries of finance, that they need to invest in pandemic preparedness. And our work primarily focuses on OECD countries, as well as EU, EEA countries, as well as the G20 countries. So it's a little bit broader focus, though we're very thankful to the European Commission for the funds that we received for the project. So a part of the work that we do is that not making the case for pandemic preparedness requires that we also think about the strategies that we can use non-pharmaceutical interventions, as we call them, to intervene when a pandemic or any type of outbreak starts spreading in the population. So this would require us to think about not only health sector specific interventions, but beyond that, what else can we do outside of the health system that can complement the work that we do within the health system? So when you think about non-pharmaceutical interventions, you can think about from our harrowing experiences of physical distancing to complete lockdowns of our societies to teleworking arrangements to other interventions such as improving the testing, tracing capacity, etc., of our communities. For us, wastewater surveillance becomes a part of this equation, part of the equation about what are the non-pharmaceutical interventions we can tap onto in the case of an outbreak. And specifically in our work, we're looking into how wastewater surveillance can help us in the context of 5 specific pandemic outbreak scenarios. The scenarios that we look into are ranging from the evidence that we collected based on the current scientific literature, going from a potential new SARS-CoV outbreak to a pathogen that can mimic a huge threat like measles. So we have a very wide range of pandemic scenarios that we're thinking about. I don't have any slides to present you some very cool results that we have, but I do promise that we do have them. But the overall messages that we see are in our data is actually very clear. First of all, we can show that we can use economic models that also combine epidemiological factors. So these combined models, mathematical models, can give us a lot of information that we can use to inform policy making. One of the issues that was discussed before is that in the case of an outbreak, one of the issues that policymakers across the world grapple with is, in fact, uncertainty. And when we think about uncertainty, we know that one of the ways that we can start to address it, not completely get rid of it, but start to address it, can be wastewater surveillance. Because we see that in our analysis that if we use wastewater-based surveillance-based information, we are able to improve the accuracy of our predictions about the potential dynamics, potential ways in which a dynamic can evolve. It's incredibly important information in the case of an outbreak because it is incredibly important for decision makers to act quickly, when to act, what to act, what kind of non-pharmaceutical intervention that they need to intervene to introduce or scale back. And we see that by improving the predictions that we use in our mathematical models through surveillance, we can help decision makers make the decisions based on evidence that is a little bit more accurate. And in the case of a fast-paced, quickly evolving situation, this is absolutely important. Though I have to highlight, we don't recommend that wastewater surveillance should replace the traditional methods of testing, of course. But what we're saying is that we can add this very valuable tool to our toolbox. Another very big result that we have is that we see that combining wastewater interventions with other types of non-pharmaceutical interventions, such as a policy like physical distancing, could help not only the effectiveness of these types of interventions, non-pharmaceutical interventions, but also it could help policymakers decide on the trade-off between economic and health outcomes. And many of you, I'm sure, have followed the discussions during the pandemic and COVID-19, is that there is this clear trade-off between economic outcomes and health outcomes. And policymakers have to weigh these decisions. They have to weigh these very sometimes contradictory objectives. So what we actually see, for instance, is that if we introduce data that we collect from wastewater into decision-making, into our models, we are, for instance, able to see that the number of days that societies might have to endure in a lockdown situation, so a complete lockdown for societies, just by simply introducing wastewater, could actually reduce. This could mean that we can go to work, the sections of the economy can remain open, or at least remain a little bit more open depending on the situation, but the pain that we have to endure economically could be reduced. And this is a huge consideration when we think about the policymaker level decision making. And finally, we are also seeing that when we think about the budget and how much it's going to cost our societies to scale up the existing levels of implementation of wastewater, I can reassure you that this is in fact a relatively cheaper measure compared to other interventions that we have to look into. And in fact, and maybe a teaser for our report, we see that for many of the countries that we included in our analysis, the per capita, per person cost of investing in wastewater surveillance is less than €1. So it's worth thinking about making this investment and to scale up the existence of the data that we have. So I'm going to stop here and then pass the floor.
Yeah, Lourdes, you maybe can have some reflection after Elche.
Well, my homework, firstly, good afternoon to everybody, and I would like to thank OBEPINE and WHO for the invitation to share the Brazilian experience, and also to salute all of you. And my homework would be to say a little bit, to describe how it is going on in Brazil, how it was built up. And so, yes, again, I will make some kind of advocacy, because wastewater surveillance, it has accumulated lots of data, lots of experience, although not integrated as a surveillance system, but on areas where we have sanitation. And our main worry when thinking about surveillance, because as a national reference center, and Fiocruz is extremely involved into public health development implementation in Brazil, because we are part of the Ministry of Health, was what about those who doesn't have sanitation? And they are the most vulnerable populations for infectious diseases. About one fourth of Brazilian population, it's about 50 million people. They are living in these conditions, and it's so common in the Global South. So how are we going to really build up a system leaving no one behind? And so as fast as COVID came, the Ministry of Science and Technology launched the call for research. And it was a very, very innovative and integrative approach in Brazil, because all research groups who had a project approved, they were organized into networks. And one of these networks was exactly the wastewater-based surveillance. And so it was an opportunity to set up the first network, I would say, but for research purposes, and where we could share information, protocols, discuss results, and even exchange reagents when necessary. And so this was the very first step to try to organize something. And some months later, the Ministry of Health started a pilot project to really train and give all support to 8 public health laboratories to establish the methods, and then to be able to be in the field to test samples. But the problem in this sense was that they were not able to collect the samples, because the sanitation is private in Brazil. So this is a huge difficulty to really set up for sewage system, but not for the sanitation area, which is lacking too. And so because of that, we decided to start by the most difficult, and then we chose a region in Rio de Janeiro that is, it encompasses the biggest slums and lots of informal settlements and so on, to really have an experience of how to try to organize that. So this region, you have a very, very hard socioeconomic situation, also along violence. And so, yes, it was the fillet and all the difficulties we could have. And so sometimes we were not able to enter the territory to make election because of violence. And in the other hand, the community was coming to take doubts, what are you doing here? And so it was a very, very nice experience. And in fact, because of that, we started developing many other activities. And so wastewater surveillance must go beyond research or even a pilot project. It must be a decision from the Minister of Health, who is the legal instance to really take surveillance actions. And after talking, because we had a difficult government and science and so on, so it was very hard. And this initiative didn't come up. We moved, the government was changed, and finally we, together with the Health Emergency Department, we are setting up a working group to then establish guidelines, looking at both on areas with sanitation, areas without, how to deal with that. And yes, then other problems come from methods, how to sample, into so distinct contexts like we have in Brazil, because if you go to the north, it's absolutely different from the south. And so how to set up some guidelines that we are able to cover all these specificities. Yeah, and that's our homework right now. So we have a group, and this group is formed by public health institutions, academics, surveillance system, to really try to set up these guidelines and really go ahead. And so this is the commitment we do have at the Ministry of Health, to really try to start something, listen to all, and then try to set it up at the National Public Health Laboratory Network, because they must do surveillance and this must be decentralized. In casing of scaling up as pandemics, as COVID pandemics, then we have our research network that is also part of this big network. So it's like a national treaty that's being coordinated by our Minister of Health to try to really set up in the best way we can the structure to make it happen, not only as different research groups creating their own results and then publishing, and that's it. The budget was finished, so it's over, but as a continuous and sustainable research. In addition, one of the biggest challenges is how to compare the results everywhere. Yeah, each group is using a different target, a different sampling, and so on. As in Fiocruz, we have one producer unit for diagnostic kits besides vaccines, biopharmaceuticals. We just sat down with them. Come on. First of all, we need to understand these results because it's totally different from clinical samples. So we have been designing and validating, right, that this is step now, a kit, a molecular panel. And the idea is from viral concentration to viral detection to have the single product that we can scale up in a very reasonable price, because we are a non-profit public institution, to distribute to all our networks. So what's going on in one state, we will train everybody, and the idea is we can compare. It will not be a bias because of the testing or the sample or whatever, but this is the real epidemiological scenery. And so if I would address our question from this session, talking about sustainability, I would resume into 5 points. First, Political decision and commitment are crucial for establishing new public health programs, ensuring that necessary regulatory framework and resources are in place. And so that's why the Minister of Health must be ahead of this kind of initiative. Otherwise, it will burn and die. Second, public reliability and equity. Wastewater surveillance can help public health officials to pinpoint communities in the need of targeted interventions, such as increasing testing and vaccination campaigns, improved infrastructure. That is extremely important. Furthermore, it facilitates a more equitable distribution of resources by ensuring that vulnerable populations receive the attention and care they need. It's a constitutional right in Brazil. Incorporating wastewater into public health strategies can enhance overall community health and foster greater equity, ensuring that all individuals have access to the essential services necessary for their well-being. In this context, communication is a key activity, as we have discussed in this morning. Third, revisiting surveillance framework, combining various logical, clinical, climate, genomic, wastewater, animal health, rumors. We have a very beautiful project in Brazil that is under course, and it's being adopted in some states and by the Ministry of Health. It's called ISOP under Dr. Manuel Barral coordination, that it's working with big data, because in Brazil we have the national databases, and so he's importing everything in modeling. It's extremely powerful and has been one of the topics we are in cooperation with the Global Hub for Epidemic and Pandemic from WHO. Besides that, global and national databases and sharing of information would enhance epidemiological intelligence at different governance levels. And so we have to start by our home and then to really scale up. Fourth, building local capacities and engaging communities, as people remain the most valuable resource and asset in addressing health threats, particularly in vulnerable and hard-to-reach areas. How to reach rural areas, how to reach groups which make illegal activities if we don't have the community beside us. So this is our day-by-day exercise in these vulnerable areas. And last but not least, one of the most important is funding. Otherwise, we don't have sustainability. So increase the financial support would enable ongoing methodological advancements and systematic long-term surveillance adapt to different pathogens and environment contexts. Such investment could ensure that wastewater surveillance become integral to public health surveillance globally, providing data that bridges gaps between countries and bolster collective health security initiatives. And furthermore, dedicated resources can accelerate implementation of wastewater-based surveillance in low sanitation regions, customizing the surveillance methods to situation where traditional healthcare system encounter obstacles. We have white maps in these regions. This people is abandoned. With greater investment, wastewater-based surveillance can evolve into a transformative public health instrument, offering timely insights and a data-driven foundation for pandemic readiness. Global initiatives such as the Pandemic Fund would serve as a strategic source of investments to really enhance this activity all over. So, thanks.
Thank you, Maria de Lourdes. It's almost a roadmap you are presenting. And you also point that Brazil is not only a country, it's almost a continent. And so it's a good transition to go to Europe now, which is also a continent. And Wolfgang, Philip, you belong to HERA, this initiative from the European Commission. Can you explain what is HERA and what can be the proposal for promoting wastewater surveillance?
Thank you very much. I'll be relatively short. I think it's always the worst slot in any kind of meeting to be on the last position, but I try to be concise. So about HERA, HERA was created basically out of the pandemic. HERA has let me read it out, has one simple mission: that's strengthening Europe's ability to prevent, detect, and rapidly respond to cross-border health emergencies by ensuring the availability and access to key medical countermeasures. So what are we doing there? We have been seeing, Chikwe, you have been referring to many of these shortcomings and problems we had during the pandemic. I will not repeat all of these things now, but the idea is really to have an entity, and we are part of European Commission, kind of policy making, but also funding. We have different hats on. What we are doing is basically we created an entity that has the function to do intelligence gathering and assessments, then development of medical countermeasures through research and development activities, supporting clinical trials, investing into manufacturing of medical countermeasures, and that's mainly obviously vaccines, drugs, diagnostics, but also non-pharmaceutical products, but also do procurement, stockpiling, and deployment. So it's the whole range of activities. And wastewater is one of the wastewater surveillance or wastewater monitoring, I have to say, is one of these activities that is in the very first complex that's around intelligence gathering and assessment. So why are we doing this? Simply because it's a tool, it's a technique that exists. It has shown its advantage and usefulness also already during COVID-19. A recently written article that described the whole history going back to 1867, using sewage water to control certain events. So therefore, that's why we're using it. How do we use it? We had a lot of discussions with WHO, with some of the philanthropic organizations, the bigger ones that have been working with us and with us. That means the European Commission, so HERA, and in particular the colleagues of the Joint Research Center. Some of them are here today. Basically on a system setting up what was earlier already mentioned, that's GlovaCon. So that's a global network for wastewater surveillance. And the idea here is not to counteract on anything that that an organization like WHO or others want to do. It is really to be inclusive. And the point is, or what GlovaCon is doing is to work on some of these elements and some of these questions that were mentioned by Chikwe in his keynote talk. So I think we are all working towards the same goal and outcome, and let's agree on all of these points which are important. That goes from governance to data to protocols and so on and so forth. Everything you need to have at the end of the day comparable data and to be able to detect earlier what's going on and observe how certain pathogens circulate, and also to have better information to model trends and contribute to decision making, because that's at the end of the day that's the final goal. I mean, and decision making comes always, that's also no secret, was mentioned a couple of times, with a certain level of uncertainty. But reducing this uncertainty, that is the final goal. And wastewater surveillance is only one of the tools that that have a function to play there. So apart from GlovaCon, we are also working in particular with EU member states through something we call a joint action that is basically supporting activities to consolidate wastewater surveillance for public health in Europe. That's a huge project where 15, 16 member states participate. No, 23, I think. So quite a number. We collaborate also with WHO. We collaborate with the UN environmental system, and we are investing a lot of money into establishing, at the end of the day, a global wastewater sentinel system. So all of this is not done in parallel, but in cooperation with WHO, with other players in the field, and in particular with the countries, not just in Europe. GlovaCon that comprises already, I don't know, about 60, 70 countries that participate in these activities. And we are just one of the players there, but with some considerable investments, we hope that wastewater surveillance will be done let's say, in a useful way and in a smart way, so that the data can be used for decision making and public health interventions in case that there's need for it, and also in case that there's no need for it, following a thorough assessment. So that's what we're doing at the European level. And I stop here, if that's okay. I could talk a little bit more about some of these projects, but I think many of you know about these projects or are even involved in many of these projects. Thank you.
Yes, thank you so much for all these comments. Maybe before we take some questions from the audience, if any, or from the remote side, maybe Chikwe, you want to add some comments to what you have heard today?
Thank you. I think my voice has been heard enough, but I do think there's, just listening to the comments, from national institutions, from, regional bodies, and we've worked a lot with HERA, benefited from your support, but also your thinking with academia, the number of is actually participating in wastewater surveillance, especially the airport-oriented one. So I think there are a lot of players in this space, which is a good thing. 30 years ago, there would have been an opportunity and the world would wait for WHO to come up with guidance and everyone would be paralyzed until it does so. Now we're in a different world, that when there's an opportunity, there are lots of initiatives driving it forward, and our role is changing, that we have to think together, respect the opportunity that academia provides, digital surveillance systems offer, countries offer, regional bodies offer, financing bodies offer, philanthropic organizations offer, and see how we can make the best use of the various skills, resource, expertise that we all bring to the table. So it's not a problem in wastewater surveillance when it's a good problem to have. We're not in a position of absolute scarcity. We're in we have a problem of making the most efficient use of the expertise and resources that we all bring to the table. And it's a humbling position to be in. And maybe that's why this meeting is so important with the players in the room.
Thank you. If you have any questions, please raise your hands because we are very talkative, so we may omit to give you the floor. There is someone, maybe the chat.
I can begin, actually. I have the microphone, so it's easier for me.
Yes, Vincent.
No, it's kind of question or comment, actually. The feeling I have is that we can easily find money for research programs on wastewater-based epidemiology in France, in Europe, or elsewhere. The Bill and Melinda Gates Foundation is providing quite a lot of money as well. But my feeling is that we have difficulties to find money for sustainable system. I mean, for prevention, basically. It has been said this morning, prevention is always difficult to finance because if it works well, you don't find anything except the day or you find something. So I would like to have your feeling on how to make a sustainable system, not just research focused, but really something that can be efficient for years.
In cold times.
Yeah.
During the cold times. Who wants to comment on that? Chikwe?
I think I've spoken enough. Let me. Colleagues, maybe in Brazil, what are you doing where you don't find anything?
Well, in fact, that's the 1 million question. Yeah, Vincent, because although we have some budget from the Ministry of Health, yes, that's if we are thinking about respiratory viruses, but the potential is too much more. And with so different realities that we have to prepare, so different regions, the budget we do have for 3 years is not enough. And so I think if wastewater surveillance has the real value it has, so it's easier to get some support. But for that, we have to show very good results. And that's it. Maybe integration is the way to really have I would say, better results. So it's working out into different realities. I think it's already proven that we have a very powerful early alert in the hands, but that's it. We must convince the founding persons and institutions that yes, it's cost benefit. Also, we must be very clear that wastewater surveillance is not coming to replace, to substitute the classical surveillance. It's coming to add. Yes, because everybody loves their boxes. And I think this is one of the main difficulties we do have to have integrated systems. But we are not going to respond to new challenges we do have enhanced by climate change, by so many other variables. If we think small or inside our boxes, this time is over. But it's good to be clear, because we will have less resistance, I would say, less people playing against, yes, when we are going to ask for funding. So the funding must come to a real response and integrated response. Otherwise, it's very hard. You'll get only small money here, small money there. It won't be enough to make a sustainable long-term surveillance as it must be done. Yes, I don't know if I was able to answer, but.
Just to add and follow up on what you're saying, which I completely agree, when we look at the global level action, ironically, I was looking at this data this morning. For instance, between 2022 to 2023, we do see that there has been a 32% increase in the aid flows that go into pandemic preparedness. That's very encouraging. We welcome even more funding. However, we do also see that while we are increasing funding for certain areas like pandemic preparedness, we seem to take money away from other areas that still needs funding. For instance, we see that approximately 2% decline in the global aid that goes into WASH interventions, so water sanitation interventions that we heavily need to rely on for pandemic preparedness. So we need to be at the global level, be very careful about what we are funding and how we are funding to make sure that our interventions, let them be WASH, let them be infection prevention and control, let them be non-pharmaceutical interventions, these programs receive predictable funding, because these types of issues that we deal with, such as pandemic preparedness or antimicrobial resistance, these are issues that are not only challenging our systems today, but they are long-term threats. So because of their long-term nature, we do need to have a long-term view on how do we find these programs. So I'll stop here.
I mean, just to confirm, basically, integration is the keyword here. Obviously, I mean, there's different levels of funding and funding opportunities. I mean, we have been at the European level into wastewater surveillance or wastewater monitoring, I have to say, but also developing genomic sequencing capacities. In new member states, we have been invested more than 100 million euros already. So it's just part of the equation. At national level, there is funding opportunities from philanthropic organizations as well. But I mean, the window of opportunity is certainly closing at one point. I mean, you're saying it, looking at some of the trends in terms of investments into what is a big block of pandemic preparedness. So therefore, it's a good tool. It has a huge potential to be integrated into public health functions, and that is where it would make a good deal and tell a good story for, let's say, moderate spending. I mean, moderate as compared to other things like development of drugs or vaccines. A moderate spending with potentially a very great impact on outbreak control.
A question or comments?
Yes, I have a question, online question from Louise Stephanie Makemge, and she's living in Cameroon. and she's wondering if there is a plan, if WHO has a plan to develop such approaches in these developing countries.
So thanks, that's a very important question. And there are actually lots of, I don't want to call them pilot projects, but there are lots of initiatives going on already. And our work now is to really think together and on how to make sense of this, because what we define as wastewater surveillance, all those three terms need a lot of definition for this. What is wastewater for us living in Paris is completely different from what is described as wastewater in many parts of the world. So there are lots of open questions to answer. And then there's a real issue on not just funding, but the opportunity cost of where we invest scarce resources and what we advise countries to do. It's not just how much money is needed to set up a system in country X, is what else could you do with that resource for equivalent or even more or less benefit if you don't use it for this. So it's important economic questions as well as public health questions to be answered. But the short answer is this is a real opportunity for the world and something we have to develop together so it doesn't increase the inequities that exist already in both science and public health. And that's a big role for WHO to play in any aspects, and we'll work with our member states to do exactly that. Yeah, let me pause here.
I'm Anders Dalsgaard from University of Copenhagen and also from the International Center for Antimicrobial Resistant Solutions. And it's a little bit outside the wastewater area, but there was mentioning of costs and by OECD. And my question is actually to OECD, but could also be to WHO, that where are you now in the process of developing economic models and tools that countries, in particular low and middle income countries, can use to calculate, to measure the economic cost in all the different senses you normally sort of calculate these on AMR? Because from the center, we can see there's really an urgent demand. And the good side of this is, as far as we can see, it's a good business case for all these countries to invest in. But the Minister of Finance need that good model that can be used at country level.
Thank you for that question. We can talk about it even in more depth than later on, but just very quickly, in collaboration with the quadripartite institutions, WHO and the World Bank, in fact, in September of this year, we have developed a global business case on investing in AMR using a One Health approach, so a multi-sectoral way of dealing with it. Wastewater surveillance was not part of this, the set of interventions that we evaluated, but the results are online if you're interested in, and we clearly show that One Health approach that involves action across multiple sectors, not just the health sector, is a very cost-effective way of dealing with a threat like antimicrobial resistance. And let me highlight the fact that a lot of the interventions that could be used to tackle AMR can actually apply in the sense of pandemic preparedness, wastewater being a very similar point, wastewater surveillance being a very similar point on this. For instance, for AMR, we could be thinking about wastewater surveillance as a way of monitoring what's happening, the dynamics that are happening in perhaps in hotspots or in the community in general. So we can use these tools like wastewater surveillance depending on the threat that we're trying to deal with. In terms of developing a tool that the low and middle income countries can use, we are not there yet, but we do have our models for antimicrobial resistance, at least, that are now working at the global level. So we are able to not necessarily limit ourselves to OECD, but we can also look into, and when you look at our results, you will see that we have results for low-income countries in Africa to high-income countries in West Pacific region. So we have been working on the extension of our models, but still we will need some time and resources and continued collaboration with our partners like the WHO to be able to further build on this.
Thank you very much. I think it's time now to close the session, and I give the floor to Sabine for thanking our distinguished guests and all the audience.
Thank you so much to the whole of you. And as far as I understood, if I may have a very short conclusion, we have to collaborate, maybe first at the European level, but it's not enough. We have to collaborate globally. And maybe first to have a standardization of our data if we want to be stronger, and then go from surveillance to action with your help. So thank you again for your brilliant interventions today and for being with us. Thank you again.
Before we are leaving, I just wanted to say a few words to, first of all, to thank the speakers and chairmen and chairwomen. You did a very nice job, and I think the day is really fruitful. I'd like to thank also the organizing committee and notably members of the 4EU Plus Alliance that have been working for more than 6 or 8 months on this project. So thank you to all of you. Thanks to Antoine Flao, notably, probably had this idea for this meeting, and I really it was and Antoine and his team made a very good job on that, and thank you once again. Gérard Croze is not here today, but he played a very important role in this, so thank you to him. And Sabine also, thank you. Sabine is directing for EU Plus for Sorbonne University, and I did really a great job to make this day possible. People have been working a lot also. I'd like to thank first of all Diane Laroche-Joubert, who's in the back, and she's exhausted because the meeting is closed. Thank you. Lilia Brimo from 4EU Plus and Sorbonne University also, Diana Kane from the Geneva Health Forum, Carolina Leb from 4EU Plus and Sorbonne University. And thank you to the Institute of Brain, because this place is nice. The room was just great. And thank you to all of you for your attention. Thank you again.
If I may, I just want to add 2 sentences. First, thank you, Vincent, for your work and this very nice conference that you offered to all of us. And I wanted to emphasize that it's our first event of the 4EU Plus initiative on the seed fundings from our 4EU Plus European University Alliance, where we started 1 year ago to launch some seed funding for the research. Especially this project is the first one which regroup our 8 universities of 4EU Plus. So Copenhagen University, Warsaw University, Charles University in Prague, University that is of Milano, University of Heidelberg, University Panthéon-Assas, and Sorbonne University, and I said it, and Geneva.
Thank you, Antoine.
And I hope I did not forget anyone. And so it's our first research project with all of our university, and we had the chance to have also WHO taking part of this conference, and also some other partners of our universities coming, and I thank you, all of you, for coming today with us, for having an even better conference today. Thank you.
Thank you.